Morning, good afternoon to everybody. My name is Tim Schofield, and I'm pleased to welcome you to the 8th Statistics IABS Statistics Workshop. It's the longest running workshop we've had, so we thank you, the participants, for your dedication to this conference. Next. Let me tell you who IABS is. First of all, we're a scientific society established in 1955. Click through all of them, Maddie. Headquartered in Switzerland, and recognized as a nonprofit organization both in France and the United States. We also have working relationships with the World Health Organization and the World Organization of Animal Health. Our mission is to facilitate communication of regulated biological products for human and animal health with several components. Next. We host conferences like the one you're attending today to both develop consensus on key contemporary issues, but in the case of the Stats Workshop, to share our efforts across industry, the biologicals industry, and statistical practices and principles. We foster evidence-based science. We're key to our conferences is that we present science, and that science be not only presented but discussed among participants. So we hope you'll participate in today's and the conferences, or they should say the workshop, through the next 4 days. And we also have our own journal, the Journal Biologicals, where we publish not only conference proceedings, but articles based on the conferences and other articles that are submitted to the journal. For speakers, you're perfectly welcome to invite yourself to submit an article based on your presentation if you should so choose. Next, please. We have, as I said, 8 workshops. The first 4 were based on statistical and data management approaches for biotech products, but we expanded after that, included vaccines and animal health products. The 5th was on CMC development and life cycle management. The 6th was on setting specifications. It wasn't a stats workshop, but a combined effort between the statistics and the biotherapeutics and vaccines committees of IABS. Next, please. The 6th in 2020 was approaches in statistical partnership. The 7th was on QBD. We thought we'd circle around and review our efforts in the statistics, CMC statistics community as relates to quality by design. And then there's today's and the next 3 days workshop, the 8th, on growing the impact of statistics in CMC. There are other workshops I've listed that you might be interested in. There's a 3rd specifications workshop, January of 23 in Basel, Switzerland. There's also a workshop on maintaining quality through reference standards, dedicated mostly to vaccines, but the principles probably apply across biologicals in June of next year. We are planning a 9th CMC statistics workshop, probably as usual in this late third or fourth quarter of 2023, and then there is a workshop on cross-learning on technology platforms, which is becoming an extremely important topic and relates to Bayesian statistics. That is, platform technologies are fundamentally supported by prior knowledge, and that can be brought into the development paradigm using Bayesian statistics. So if you're interested, please visit the IABS website, simply iabs.org, for those upcoming events and others. There's a projects list that you might want to take a look at and show interest in if you so desire. Slides of previous events which require membership, but if you want to join IABS. And with that, I thank you, and I introduce your co-chair, Julia O'Neill, who will commence the conference. Thank you very much.
Oh, thank you, Tim. I'm very pleased to welcome you all to this conference. I've been to quite a few of these 8 over the years and found them to be great sources for keeping up with advances in CMC statistics, and also particularly in the interactions with regulatory and statistics. And this year I've worked with Christy Griffiths. It's been a pleasure actually for the last 2 years. Christy and I have worked together to set up the agenda for these meetings. This year we are taking a particular focus on extending the reach of CMC statistics and keeping it advancing along with advances in technology and CMC. So I'm about to hand it over to Christiana Kampa to introduce our keynote speaker. Just a couple of reminders during the meeting today, during the talks. If you have a question, please put those in the chat. We will be taking questions at the end of the keynote, and we will have panel discussions and breakout sessions later in the day, where you'll have even more of a chance to interact live with the other participants in the meeting. So thank you all for attending, and Christiana, I hand it to you.
Thanks, Julia, and thanks for the introduction. So it is my pleasure to announce our keynote speaker. Her name is Valentina Bernasconi. Valentina is currently the head of the Laboratory Science Group at the Coalition for Epidemic Preparedness Innovation, CEPI, and she is leading the CEPI Centralized Laboratory Network for immunogenicity testing of COVID vaccine candidates, as you will see in a few minutes in her presentation. At CEPI, she is also preclinical and immunology expert in vaccine development projects for a number of diseases. And she holds a master's degree in medical and pharmaceutical biotechnology, a PhD in immunology, and more recently, she also earned a master's degree in global health policy from the London School of Hygiene and Tropical Medicine in London. So, you know, the reason why we were thinking about inviting Valentina is that we really wanted to give you the opportunity to have an overview of global health initiatives to accelerate the development of vaccines against pandemic threats, so that they can really be accessible to all people in need, and provide evidence on how statistical tools can be of critical help in this context. For immunology testing, bridging studies to set correlate of protection for vaccines. But Valentina will also reflect on similarities and differences between immunology and CMC testing. So I really would like now to hand over to Valentina, and I remind you to keep your questions for the end of the presentation, either in the chat or live. This will be the opportunity that you will have to understand more of what Valentina and CEPI are doing in this space. Thank you very much, Valentina, for joining us.
Thank you, Christiana, for the nice introduction, and thanks to the organizer for inviting me to speak today. So let me see if I'm more lucky and I can share my screen, and I will just turn my video off so that the audio is best. So let me know if you see my slides, and I will try to put them in presentation mode.
Yes, we can. Thank you.
Okay, wonderful. All right, so as Christiana mentioned, I am not a statistician. I'm an immunologist, and today I would like to touch upon some different topics today. So I work for CEPI. I will tell you more about the organization I work for, and also the approach to assay standardization that we had via the creation of a network of laboratories, and how this could help the identification of correlates of protection for different vaccines, starting with the example on SARS-CoV-2, and knowing that this network will also expand to new pathogens in the future. And I will also try to provocate you a little bit and show you how this effort could relate to CMC as well. So about CEPI. So the Coalition for Epidemic Preparedness Innovation was created in 2017 in response to the West Africa Ebola epidemic. So as a result of a consensus that coordinated an international effort would be needed to develop and deploy new vaccines to prevent future epidemics. So CEPI was launched in Davos in Switzerland by the governments of Norway, India, the Bill and Melinda Gates Foundation, the Wellcome Trust, and the World Economic Forum. And CEPI is really a global coalition that is made up of public, private, philanthropic, and civil society organizations with the mission to accelerate the development of vaccines against emerging infectious diseases, but also enable the access to those vaccines to affected populations, so to whoever needs it during outbreaks, in order to create a world in which epidemics are no longer a threat to humanity. And to do this, CEPI funds different vaccine development projects for different priority pathogens, but also cross-cutting enabling science activities all along the vaccine development process, so from discovery all the way up to the delivery, stockpiling, manufacturing, and distribution of those vaccines. So in addition to COVID-19 vaccine development that I mentioned before, CEPI is also targeting other pathogens such as Ebola, Lassa virus, Middle East respiratory syndrome coronavirus, Nipah virus, Rift Valley fever, and chikungunya viruses. And all those pathogens are listed in the R&D blueprint list that WHO makes available every year. CEPI also invested in technology platforms that can be used for the rapid deployment or the rapid development of vaccines, and also for immunoprophylactic development against unknown pathogens, which we call Disease X. I have a link on this slide so you can see, or you can find the full list of all the vaccine candidates that we fund at this link on the CEPI website. I will give you some background and an example on SARS-CoV-2 and how CEPI approached immunological assay standardization. So as you might know, the sequence of SARS-CoV-2 was published the 11th of January 2020, and this triggered a very big effort and R&D activity to develop a vaccine against this virus. So at present, more than 400 different vaccine developers are active worldwide to develop a vaccine against COVID-19. Some of them already reached the market and some of them are still under development. But you can imagine how comparing immune responses that are induced by the different vaccine candidates is extremely challenging. Those vaccines are at different stages of development. They use different technology platforms, and the samples are collected, transported, and stored in different ways. So the standardization among different assays and different testing laboratories that is lacking makes comparison very difficult. So to improve the immunological standardization among assays and have results that are comparable, CEPI, WHO, and the National Institutes for Biological Standards and Control have worked together to develop and make available to everyone some reference serum panels and also the WHO International Antibody Standard for SARS-CoV-2. So this antibody standard is made with serum from convalescent patients, so patients or subjects that have been infected with the virus but recovered. So they're meant to have high antibody standard. We have done this kind of work also for other pathogens such as Lassa, and the international antibody standard for those pathogens is now available. So this is, let's say, the main effort for towards immunological assay standardization. But in parallel, as a synergistic approach, CEPI has also created a centralized laboratory network. So a network of labs worldwide, and we have selected laboratories with the highest quality standard. We have selected a core set of immunological assays that are key for to evaluate the vaccines, and also we have harmonized protocols and key reagents across the labs in order to reduce inter-laboratory variation as much as we could. I will tell you more about this network of laboratories. So the CEPI centralized laboratory network is open to all vaccine developers. So we offer this as a service. All vaccine developer can come and have their phase from preclinical studies up to phase 3 clinical trials. They can have their assay, their samples analyzed. And this is really to facilitate the rapid evaluation, approval, and dissemination of those vaccine candidates, and also support their pathway towards licensure. So increasing the chances that we will have more vaccines out there, and we will reach also the population that do not get any vaccines at the moment. So the network today counts 10 laboratories across the globe. 3 of them are in North America, 4 in Europe, and 3 in Asia. So we are also trying to recruit more laboratories, and we will have more labs by the end of this year, especially in the Africa region and Oceania region, because you see, we don't have any labs there. So all the labs in the network are performing ELISA. So they're measuring binding antibodies against the spike protein of the virus, the receptor binding domain, or the N protein. They're also performing neutralizing antibody assays in form of pseudo-typed virus neutralization assays, so using pseudoparticles instead of the live virus. So this is an assay that can be run in BSL-2, biosecurity level 2. But they also run a wild-type virus neutralization assay. This is a colorimetric assay. It has to be run in BSL-3. We do also have one assay to measure T-cell response. This is a very simple ELISpot measuring interferon gamma and IL-5 against a peptide pool that covers the whole S protein of the virus. So those assays are qualified and validated, so it means that can be used up to phase 3 clinical trials, and they have been developed in 1 or 2 of the laboratories and then tech transfer to all the other labs. The results from the binding and the neutralizing antibody measurements are bridged to the WHO international standard that WHO and IABS have prepared, and the results are expressed in international unit. So this allows a broad comparison of results across, not only across the network, but also with other laboratories that are also using international standards. So all the assays available in the network are based on the original Wuhan strain, also known as Victoria strain. But for the wild-type neutralization assay, we're also adapting it to test against variants, so alpha, beta, gamma, delta, and omicron. As I said before, so the CEPI centralized laboratory network is really open to everyone to test against, to measure antibodies, binding antibodies, neutralizing antibodies, and T cells. And we also do monitor new variants for SARS-CoV-2 that might come in the future, and we will implement the testing within the network as well. So the tech transfer process was developed to ensure harmonization across all the laboratories of the network. And some of the elements of success that we had within the tech transfer process were, first of all, the use of key reagents that were produced by the same source and distributed to all the labs equally. And when I speak about key reagents, I mean, for instance, controls, coating antigens for ELISA, or the live viruses, or the pseudoparticles. We also do have a very thorough tech transfer process. We start with an assay overview and teaching the labs how to reading through the SOP and touch upon the most critical points. We do go through a gap analysis to highlight the differences between the reference lab and also the recipient labs and mitigate the impact of the gaps in terms of, for instance, different instrumentation. We do have a step where we go through a laboratory comparison assessment to ensure that we have comparable results between or across the laboratories. The labs also performs an assay evaluation. This is done at the recipient lab, and this is to ensure that the assay performance is not impacted by the transfer. So the labs measure the precision, the linearity, and the specificity of the assay. And to mention, we do plot QC charts, quality control charts, and also we run proficiency assessments to make sure that the assay performance is constant over time. The labs go through online trainings and ad hoc teleconferences. One very important thing is fast and transparent communication, not only between CEPI and the developing lab, but also between CEPI and the recipient labs and the labs between each other, to make sure that all the participants of the program are really aligned of what we want to do. And of course, very important is the great teamwork that we have experienced in the last couple of years. So this slide really summarizes some of the achievement of the network so far. So this network was set up at the beginning of 2020, so when the pandemic started. So we have 10 laboratories worldwide. We offer 6 different assay types. We have engaged so far more than 50 different vaccine developers worldwide, and we have allocated more than 60,000 samples for analysis. So more is supposed to come. The future plan is really to use this network to expand not only geographically, but also to include the testing for more pathogens. But now let's dive a little bit deeper into what correlates of protections are and why our effort could help in finding correlates of protection for SARS-CoV-2. So we know that vaccine-induced immune responses are often very complex. So they're not due to only one factor, but the single component, such as the antibody response, might correlate with the level of protection against a certain pathogen. And most of the currently accepted correlates of protection are based on antibody measurements, mainly because they're pretty easy to measure, and usually vaccine work inducing antibody response that then protects against either infection or severe disease or mortality. So yes, as mentioned, correlates may differ depending on which endpoint we use, such as we want protection from infection, do we want protection from disease, from severe disease, or from mortality. And in the case of SARS-CoV-2, it's very important to connect how neutralizing antibody response to the virus works and connect this with the vaccine efficacy. And this will bring researchers and clinicians really closer to have a correlate of protection against COVID-19. So having a correlate is really important, a correlate of protection. And this will allow the licensure of new vaccines because it will be possible, based on immunological readouts, to the fast approval of new vaccines, but also it will help healthcare providers to manage vaccinations against high-risk groups, such as immunocompromised individuals, more efficiently, such as by recommending a booster vaccination if the antibody response is too low, or recommending a non-pharmaceutical intervention for protection if there is no immune response detected because their immune response is just not there. But so how the centralized laboratory network is or could help in establishing correlates of protection? Well, we essentially contribute in this, really collecting a lot of data, both via the assay transfer process and the clinical testing process. So we do collect all those data in a database, and I will tell you more about this in a second. So as I mentioned before, the tech transfer process is really thorough. We do have a protocol that has different steps, and those steps are outlined in this slide. So the first big step includes, the preparation of the tech transfer. We have a qualification phase, and then a final certification that will allow the labs to be approved to start testing clinical samples. So the preparation step really deals with the assay overview, with an initial training, gap analysis, as I said before, also drafting laboratory-specific SOPs, do some practice run and mock runs to make sure that the assay works properly in the receiving labs, and also that the controls are within the expected range, and then the labs will finalize their own specific SOP. During the qualification phase, then we do a lab comparison assessment to ensure that we have comparable results across the laboratories. And also the assay performance is measured. So the laboratories will measure precision, linearity, and clinical specificity. And in the end, the labs will write a qualification report. This is then approved, signed, and then they will start vaccine testing. And as I mentioned before, quality control charts and proficiency panel are used throughout all the process. Specifically, the proficiency panels are run every 6 months. But let me show you more in details how the qualification part is performed. So initially, during the interlab comparison, each lab will run 19 positive controls, so 19 positive samples, and 1 negative control. And each run is repeated twice. Then the lab will measure precision, and then they will measure 16 mixed titer samples in duplicate 3 days for 2 analysts. For the linearity, they will measure high, medium, and low positive samples at either 4 or 6 times dilution, twofold dilutions in duplicate. And then finally, they will measure specificity. So they will measure they will analyze 20 positive samples and 10 negative samples. So when they have completed all the testing, the results from those testing will be compiled. A final qualification report will be written. So this is done at the receiving labs. Additionally, the labs that do develop the assay have some more measurements to perform. So they will basically determine the limit of detection, the limit of quantitation, which is the conversion factor to international units. If there is any interference, the antigenic specificity, and also the robustness of the assays. So all the samples that I'm mentioning here are coming from either vaccinated or convalescent patients. So you understand that the amount of data that we collect on immune response against a certain virus or on the immune response that elicited by a vaccine is huge, because you need to multiply this by 10 labs, 6 assays, and so on. So this is an example of results for a linearity study for 3 different assays. So you see S-ELISA, N-ELISA, and RBD ELISA. And you can see the expected titer and binding expected titers on the y-axis, and you can see also that we get a good linearity within each lab that are presented on the x-axis. The difference between labs is greatly reduced when we normalize against the international standard, and the R2 is consistently greater than 0.99. These other slides show you the correlation between MNA and PNA. And you can see that there are 2 ways to measure antibody neutralizing antibodies. So we have an MNA, and MNA means that is a neutralizing assay that uses the live virus, and the PNA that uses the pseudoparticle. So let's say a non-live virus that expresses on the surface the proteins coming from the live virus. And again, as you can see, the 2 assays correlate pretty good. So as I was mentioning before, to keep track of the assay performance over time and also across laboratories, we do run proficiency testing every 6 months, and we plot the data on charts. And for each assays, the labs runs standard, different positive controls, and also the results need to meet strict acceptance criteria. So we do this every 6 months. So we do collect all the data coming from the tech transfer and the proficiency testing, but also, of course, we do collect data coming from the clinical trial. And we collect them in a database. So we have developed a database to store all the data. Those data are automatically pulled from template forms into the database. So this means that basically when a laboratory is running their study, they insert the data in template forms, and they all look the same for all the labs, so that the program will automatically pull the data from those forms. They also undergo a series of automatically quality checks. They are clean, they are processed, and then they're used for further analysis and reporting. So now that I show you how the centralized laboratory network works, we can get to the final part of my presentation, and I will try to identify some similarities and also some differences between our work, so immunology basically, and the CMC field. So the immunological assessment and the CMC testing have in common for sure the need of comparing results across vaccine technologies. They also have to deal with the use of standards. of standard assays, which unfortunately sometimes have low throughput. And they also both benefit from the use of international standard and the use of statistical tools for assay comparison and tech transfer, for example. On the other hand, CMC probably is more focused and more looking forward to try to avoid the use of animals, while in the immunology field, we still rely on testing vaccines on animal quite a lot. And also CMC focuses more on assay specification. And for CMC, perhaps the measurement of the vaccine or the measurement is vaccine technology specific. So you will need to measure parameters if you have mRNA vaccines for COVID, and if you have a viral vector based vaccine for COVID, you will need to measure all your parameters again. And this is not really the case for immunological assays that go beyond the vaccine technology per se. One last thing to mention is that also having correlate of protection and a link to preclinical studies could be useful for consolidating vaccine characterization studies, for example, to confirm which structural characteristics have an impact on the efficacy of a vaccine. And the in vivo assays are often used to test product target compared to structural variants to understand if there are differences, and then to define the CMC analytical strategy. And for example, if the variance is less active than the target antigen, it for example must be ensured that in that specification and a chemical physical test to monitor that the presence of these truncated forms is needed. So it would be really important to have a link with the preclinical testing and the clinical serology output or correlates of protection to have more reliability, or to give more reliability to CMC study. And it would also allow to propose for specific ranges for these truncated forms. So just as a conclusion, I would like to throw a question at you, and a little bit of a provocative question, and see whether you think that the approach that we had or that CEPI used with the centralized laboratory network could be applied to your field as well. And with this, I would like to end my presentation, thanking all the labs that participated to this effort. And of course, thank you very much for your attention. And now I'm happy to take any question. And I hope I will meet the expectation and you will not kill me with the statistics.
Thanks very much, Valentina. Thanks. That was super interesting. Really, really thanks for sharing these efforts with you. And also thanks for all the work that you are doing along with all the collaborating laboratories for this emergency and for pandemic preparedness. All right, so we can now open for first some questions, from the audience, but maybe I can start responding to your question just to begin, and maybe others can comment. So I think that many of the elements that you mentioned, especially in the last slide, can actually be applied to the CMC space. Not only the statistical tools, but also really enabling more connection actually between CMC and clinical elements. And you made very interesting examples talking about structure-function relationship understanding, but maybe also thinking more really on what could be ranges for critical quality attributes, having such a strong knowledge on the immune response. So I think it's definitely a way to go. The point I see, which maybe goes to a first question from my side, is that this was really applicable, or is being quite successful, I would say, based on your data for COVID, where you had really a lot of samples, a lot of institutions and companies willing to share really to fight the pandemic. But I'm wondering, in a normal scenario or in an epidemic scenario, because I think you would like to use this network also for future pandemic preparedness, what would be the issues that you see and the elements that you could use from your experience for other pathogens?
Yes, and that's a great point. And the easiest answer would be that, so in the case of COVID-19, we have 10 laboratories and we will have 16 very soon. So all of them use the same assays and all of them do clinical and preclinical testing for SARS-CoV-2. We do work on other pathogens, as I was showing on the slide, such as Lassa virus or Nipah virus. And you can imagine we don't have 400 vaccine developers trying to make a vaccine against Lassa. First of all, because there is no market for it. So, and we are funding 5 of them, if I'm not mistaken. Maybe there are other 2 or 3 out there that we are not funding, but that would be about it. So the easiest part there would be that we will not have all the 16 labs, doing assays, because there is no samples to test. So we will divide the laboratories in groups, and each group will take care of one disease. So that's the easiest answer. What I really hope is that what we have learned and what we have experienced with the pandemic, so basically the sharing of results, sharing of information, will not stop after we are kind of done dealing with COVID-19. I really hope this will be something that we will bring with us moving forward, and use on other pathogens as well. I think it was one of maybe the few good things that came out from the pandemic, and I really hope that this will be brought with us. On a separate note, so CEPI is dealing with 5 priority pathogens, and those are endemic in some areas, and they have been chosen because they have epidemic or potentially pandemic potential, so they can create problems in the future. But one thing that we are really looking at is disease X, so a pathogen that is not, or we don't know about at the moment, but can create a problem in the future. And having assays up and running and having a framework up and running will help us do some shortcuts when or if a new pathogen will appear. So having the legal framework in place, it's really a simple thing to do in advance. But if you need to do it while the pandemic is raging, is really gonna cause you delays. So we will really leverage, we would like to really leverage on what we have created, and moving forward, be more ready and use perhaps technologies that have higher throughput and can be adapted to different pathogens in an easier way to be prepared, and shortcut the timelines of assay development and tech transfer in the future.
Thanks very much, Valentina. Very, very, very good point. Okay, so we have Tim, and then a question from the audience in the chat. So Tim, you want to go first, please?
Yes, thanks, Christiana. And Valentina, I agree with Christiana. Excellent presentation. And I saw, and I'm hoping that the rest of the audience saw, many parallels with CMC assays, not the least of which are the approaches taken for the validation. But one of the most difficult problems we face in CMC, and I also see in clinical assays, is replacing the more variable assays, such as in vivo or animal assays, with an in vitro replacement. And in the case of the clinical assays, and in particular for COVID, replacement of neutralization assays with the binding counterpart. You showed plots that indicated that there was extremely good agreement between the 2 technologies. What, in addition to showing that the agreement between the 2 technologies, needs to be shown to convince all regulators? And I would admit that there may be some regulators who agree with the replacement, but there are very hard and fast rules with other regulators that you not replace the original more, say, biologically specific assay with the in vitro counterpart. What would you advise, in addition to plots and analyses like that, to share with regulators to make the replacement happen?
Yes, definitely. And I agree with you that probably some regulators will still not like the approach very much. For us, neutralization assay is really the gold standard, and regulators like it, and we kind of, we don't have a way to jump over it for now. Showing correlation with the binding assay for sure will help us. Another thing that we're thinking of is, to go with surrogates. So basically some assays that will not use live cells or that will not use the live virus. This will help us a lot because we're dealing, specifically for us, we're dealing with pathogens that needs to be handled at BSL-3, so biosecurity level 3 or biosecurity level 4. And running a neutralization assay in biosecurity level 4 is not something that you look forward to. It's slow, it's dangerous. So we're looking into some technologies that could potentially work as surrogate assays, and they are high throughput, they are more versatile. So definitely, and I would assume that also with the on the CMC assays, you can, you know, try to look for assays with higher throughput and avoid in vivo studies. But yes, there will be probably some data to show, especially to regulators, in order to say, okay, I can go with these assays and let this one aside.
Well, the point is the statistics do show that they're replaceable. The problems may not be statistical. They may be something else we're not thinking about. And we all know the advantages. I mentioned that the in vitro are less variable and therefore more dependable than the, say, in vivo or even the neutralization assay. But there seems to be, and maybe it's a just cultural question, that is, as you say, that the regulators are used to the gold standard and they don't want to deviate from it, regardless of how useful the alternatives might be. But that you know, that is the problem I see we're facing.
And I have to say, we have been having some conversation with FDA and EMEA about this, and we will continue for sure moving forward. And the plan is also to reach out to other regulators that are not, let's say, those main, to in India, for instance, or in Africa. There are emergent regulatory agency that, you know, could be part in the conversation as well.
Thank you.
Thanks.
Great. Thanks, Valentina. So we have a question from Stan Alten in the audience. It's about, you know, what are the criteria for showing comparability at the end of tech transfer? In other words, what is the standard for comparing each lab?
So other than, you know, measuring all the parameters for the assays, what we determine from the beginning, or what the developing lab determines from the beginning, is an acceptance range. So whether if all the results coming from the labs during the interlab comparison fall within the range, then they're accepted. The range is quite narrow, so they don't have so much you know, room for errors. And what they will need to do is really to repeat the assay until it really gets within the range. What we have realized, one thing that we didn't think from the beginning, when you give a developed SOP to laboratories, and those labs are experienced lab, and they have run ELISA neutralization assay for ages before we even came with the SOP for SARS-CoV-2, but they have run it in their own way, and they have the tendency to kind of adapt the SOP to their own way of working, and this can be an issue, because really they really need to stick to the SOP that are given, and already the fact that they have different instruments, different let's say not key reagents, so PBS, plastic plates, and so on and so forth, it already impacts the interlab variation. So really following the SOP, it's a key point here.
Thanks very much. So there is another question again from Stan is thanks for the response. He is very happy with your feedback. So I have a question from Jose Ramirez. You mentioned the ranges are narrow. How are the ranges developed?
Okay. Developed. So yes, so this is developed at the developing lab. So they do all the work of assay development. So they optimize, they take a normal ELISA SOP, they optimize it with the antigens and antibody, for instance, that they need to use for the specific pathogen. And then they have to determine which is the cutoff of the assay, and they will then use standard deviation above and below, and to determine the range. So it's usually less than 1 standard deviation.
May I add to that question? I noticed that you don't, we're talking about range. I noticed that you don't determine an upper limit of quantitation. Yet that's required in clinical assays that I'm familiar with, which would make the range. And could you comment on the fact that you don't actually report a range? That is, you don't report an upper limit of quantitation. Is it that the regulatory authorities you're familiar with don't require that, or?
No, they do. I think I missed that on the slide.
And maybe the answer to Jose, I just happened to be writing a paper about this. And there is a way of determining the range of the standard curve using an uncertainty function, or the delta method, Jose, which allows you to declare where the the region in the curve that that reports adequate precision. I'm not saying this to say anything negative about Valentina's, but just as a discussion among the statisticians here. There are tools for answering that question that we have at our disposal in mathematics and statistics, which are quite valuable, and it gets around an issue that we sometimes believe we have, which is by testing samples, we're not really testing the range. That is, we're only testing the range among the samples that are being tested. We're not declaring or determining the range, the analytical range of the assay. So there's complications also with expectations for determining a range that can be overcome using stats and mathematics.
And perhaps if I can add to what Tim was saying, one of the problems that we had, which I'm not sure if you have it in the CMC field, once when those assays are developed, they are developed sometimes before any vaccine is available. So the samples that are used are samples from convalescent patients. So their antibody response is limited, and it's basically the one of a person that has been infected with the virus and recovered. But when we then test samples, we test samples from the vaccinated people, and most of the times their antibody response is much higher compared to the one from the convalescent patients. So what the labs have to do when they go to test the clinical samples, they need to change and change the ranges. And the one other issue that we have is that at least for those assays, I'm not sure for other immunological assays, but for the one that we have, the part of the curve that is linear is quite narrow. So that's something also that we might want to look into and see if we can solve somehow using other assays.
Thanks all for the good conversation, Jose. Hopefully this addressed thoroughly your question. Yeah, indeed. And
yes, it does. Thank you, Tim. Will be something to discuss later on. And thank you, Valentina. Very interesting.
And sorry for my poor statistical background.
Oh no, not at all. This is very helpful for all of us.
Do you have statisticians at CEPI?
We do actually, luckily.
Oh, good. Glad to hear it.
Yeah, and indeed, this links to a question from Julia. Really, you mentioned, Valentina, normalizing results to a standard to achieve greater consistency between laboratories, right? So our statisticians involved in this and other strategy for comparing results across laboratories. So can you confirm, what you just said and expand maybe a little bit on how you engage with the statistical experts? Thanks.
Yes, that's correct. As I was mentioning, luckily we do have statistical experts within CEPI, but especially at the labs. So they are the one running the main, statistical analysis. We follow the process and we give support if needed, but really the big lift of the work is done at the labs. regarding the normalization and comparison to the normalization to the international standard is something that NIBSC and the WHO do routinely when they develop the international standard. So what happens there? They collect serum from convalescent patients. They choose the one with the high antibody standard. They pull it, and then they say, okay, this is the standard preparation. They then send this preparation to different labs across the globe, and they will all use their own binding antibodies assay and neutralizing antibodies assays. What they really want to see is that this preparation is working across all sorts of assays and in all settings in different laboratories. When they have made sure about this, then this preparation will be called international antibody standard. And we do more or less the same thing. So we use the same reagent. We also have an internal secondary standard, we call it, because simply because the international standard is limited in the amount. So we do have our own internal standard. We calibrate it against the international standard. So basically we test in the same setting and we come up with a correction factor. And then from that point on, we will be able to express all the results that we have in international units.
Great, thanks very much. And just a quick comment on not being a statistician. I'm not a statistician, and I was in the committee of this workshop preparation. It was a super interesting experience. So I hope that you will be more and more exposed to your statistical colleagues. They can help really a lot, I think, this
Didn't
discussion.
you volunteer to do Bayesian analysis, Christiana?
Good question. I'm not able yet, but if you teach me, maybe I can give an attempt in a few years.
We'll be happy to help.
Good. All right. So is there any other question? Because I have another one from my side, but I don't want to block others to I don't see anything in the chat at the moment, so maybe I can go with mine. It's a more provocative questions, and I don't know if you can answer, but you are talking about correlate of protection for COVID that will be available. Do you have now confidence of being close to a correlate of protection? You shouldn't say, of course, what it is, but we would like to have a guess on where you think we are overall, because I think you have a broad view on that.
I think we're closer than we were probably a few months ago, but probably not there yet. So one complication in general, not for COVID only, is if you have other components of the immune response that have an impact. So I'm thinking, for instance, for Lassa, neutralizing antibodies are not really the one that do the job. T cells seems to be more important, and measuring T cells is a little bit of a nightmare compared to a standard ELISA for antibodies. For SARS-CoV-2, we have a lot of data out there. Not all the data that I've seen are in international units. So again, comparing is very difficult. Data coming from the developers that have used our network will be in international units. So one thing that we do from our side is really pushing them to publish, because as a CEPI per se, we don't own those data. We are not allowed to publish those data on behalf of the developer. Is really on their side to do that. We will be able to publish some anonymized summaries. So this maybe will help too. But I'm afraid we're not there yet.
Thank you. And so what I'm hearing is that, it's more complicated than just antibody response first, plus you are still having some analysis of data from with different units and different sources. So these are the two main points, right? Thank you. Tim.
Well, to that question, isn't the fact that there are variants also a problem? So
Yes.
again, we hear that the different variants have different manifestations in the disease. So when you talk about correlate protection, it's there's different definitions of the degrees of disease, and also the different variants appear to have different properties as far as disease severity or progression. So is a correlate of protection even, well, in addition to the other issues, is a correlate of protection even a goal when you have a virus like a flu or or COVID, where the disease is different from variant to variant?
That's a very good point. And to add on that, they not only have a different manifestation in humans, they behave different in the assay. So that's really a nightmare. Every time that we have a different variant, we need to re-optimize the assay, especially neutralization with live virus. I think that's the most critical one. In terms of ELISA, not so much. The mutation that characterize one variant compared to the other do not impact ELISA so much, but the live neutralization assay it really differs. To up to the point that at some point we cannot use colorimetric readout, we have to switch to fluorescence, and then you have a completely different assays, how you can compare really. And on top of that, the international standard was made with convalescent serum from the original outbreak, so the one strain. Now there is a replacement coming up because they ran out of the material from the first standard, and these will include some variants as well. But again, if in 2 months or 3 months we have 2, 3, 4 more variants, then we are kind of behind again.
Oh, thanks. It's not easy at all, right? It's but it's a great challenge, I think, right? So the bigger is the challenge, the more interesting is to work on it, right? So it's thanks for working on it again. So is there any further question or comment for Valentina, because we are approaching more or less the end of the session?
That's it.
No?
Oh, I just wanted to chime in and say that how much we appreciate you sharing your perspective on this assay. And please don't apologize for not being a statistician. One of the key features of CMC statistics that I think sets it apart from applications of statistics in other areas is how important the context is for the application of statistics. And for CMC statisticians to really develop a high level of skill means that we need to become knowledgeable in the areas where we're applying statistics. And certainly never to the level that, Valentina and Christiana, that you are in the assays. But the more we learn about those, the better we can partner with you all on the strategies for improving assay performance, and certainly in this case, comparing across laboratories. It's actually an area that was done quite commonly years ago when I worked in the chemical industry, and is a bit less common, at least in my experience, in pharmaceuticals and biotech, but I think there's tremendous value there. So Christy and I had made the decision to reach out for keynote speakers who were not necessarily statisticians, but people who can help us all learn about such an important area of application. And certainly things related to COVID have been subject of great interest, and for the last few years was very relevant. And thank you so much.
Thanks.
Thanks very much, Julia. And I think that then we can now say thank you again to Valentina, and maybe move on with the next session for the agenda. So Julia, I'm handing over to you for the introduction of the next session. Thanks again, Valentina, and thanks
Thank you very much, everyone, and enjoy the rest of the conference.
Thank you. Thanks. Bye.
Okay, well, we got a minute. Hoping everybody's trickling in now. We're going to get started in less than a minute. Okay. So welcome everyone. My name is Joanne Coleman and I am the Research and CMC Statistician here at Spark Therapeutics, leading a small group supporting everything from discovery straight through manufacturing at Spark Therapeutics, which is a gene and cell gene therapy company that was spun off from CHOP. And we are now a part of the Roche group. Welcome to session 1. Thank you to Valentina and Christiana for a great keynote this morning. Session 1 is going to focus on how we apply CMC statistics in new modalities. And we have 3 great talks today. And Jose Ramirez and I are running this session. We'll be monitoring the chat. If there are any questions during the talks, you can throw your questions in the chat. And of course, there'll be a panel discussion at the end of all 3 talks. So first up today, I'd like to introduce Rick Burdick. And he is from Burdick Statistical Consultant. And Rick is going to speak today about statistical approaches for CMC comparability testing for cell and gene therapy. And I'll let you take it away. I don't know if I'm supposed to ask if you want Abigail to run the slides or if you want to run them.
I'll run it myself. Yeah, I think I can
Great.
share the screen then and do that.
Okay. Thanks. Welcome, Rick.
Thank you. Welcome everyone. I will share my screen here. And if someone could just ensure that they see my
We can see it.
see the screen here.
Yeah.
All right.
Can you put it in presentation mode? Will be better.
Yeah, I will.
Thank you.
Thanks. Yeah. So I get it going here. All right. How's that, Jose? All right. Thank you, sir. Welcome, everyone. Just one observation before I get started. It always appears to me when someone tells me they're not a statistician, they have a smile on their face as if they've been blessed somehow. And I don't know, it's just an observation I've made, but my lot in life is to be a statistician. So that's what I'm gonna talk about today. I recorded this talk a couple weeks ago and I ran out of time. I was trying to talk about too much stuff. So I'm going to focus on one particular example today. And in particular, what I'm going to do is for a demonstration of comparability using an equivalence test in a split apheresis study, which is pretty particular to the CAR T cell products. And in terms of work I've done with clients, it seems to be a design that the agencies like to see. So I want to talk about some of the issues that come up with this particular technique and take you through a couple of examples today. So at a very high level, as most of you are probably aware, CMC comparability studies, the early guidance from FDA and the ICH Q5E noted that changes to manufacturing processes are needed in order to improve our products, but we need to ensure that any changes we make don't somehow adversely impact either safety or efficacy of our product. So what we're forced to do here, or what we're called to do here in a comparability study is compare both pre-change and post-change processes in some manner to ensure that the post-change is close enough to the pre-change that we don't have any unintended consequences as it relates to safety and efficacy. And in terms of my tables and pictures today, when I talk about the pre-change, I'll call that the reference process, and I use the letter R to denote that. And for a post-change, I'll call that a new process, and I'll denote that with N. So again, we have a host of applications that we have here. And what I'm going to look at is the comparison of a CAR T cell product after we've made changes in the manufacturing process. And the way this design is carried out is apheresis is performed on a sample of healthy donors typically. I'm picking the number 6 here for this example, and we're going to look at, one of the things I want to show is the impact of the number of donors on the ability to claim equivalence. We're going to take a look at power curves, but in my example here, I'm just going to use the number 6 here, which is in some cases a typical number that you might see. So we have 6 healthy donors. Collections are made from the donors and split volumetrically into 2 halves. One half goes to manufacturing process A. I'll call that my reference group. And then the other half is processed through manufacturing process B. So again, those of you with statistical backgrounds recognize this as a match pair design or a randomized block design. And one of the characteristics of this, and one of the reasons why blocking is sometimes efficient, is that analytical measurements that we make on the resulting drug product from the 2 manufacturing sites are correlated by donor. And what I mean by that is values on one donor are more similar than values across donors, across different donors when we have a high degree of correlation. Now, what turns out to be the case is the ability of this design from a statistical sense to be as efficient as it can be depends on the degree of correlation. The higher the correlation, the more efficient the design. And correlation is the result of the percent of variation due to donor. So we have 3 sources of variation that I'll be talking about in this experiment. We have the manufacturing donor variation, which is really one of the attributes we want to compare. How do the manufacturing variations differ? We have the analytical, which is generally the same because we have the same analytical instruments. And then we have donor variation. Okay, so these 3 components make up the total variation. If the majority of that variation is due to donor, then this particular sampling design here is very efficient. What that means is for a given sample size, a given number of donors, you increase the probability of demonstrating equivalence. That is, the power increases as the donor to donor correlation goes up. So here's a picture of the design schematically. Each donor, we select our sample, we split it, and one split goes into A, one split goes into B. We do this across all 6 of our donors. Okay, now here's a plot to kind of visually demonstrate when I talk about correlation, what I mean. So I've got 4 scenarios here. Across the top, scenario 1 and scenario 2 are cases where I simulated data that have exactly the same average for the attribute, okay? On the bottom, I simulated data so this particular attribute, its cell viability, increases from reference to post-change. Over here, there's an upward trend from reference to post-change. Again, scenarios 1 and 2, they're relatively, if I take the average on the left, it looks to be relatively the same as the average on the right, okay? Now, what differentiates the blue plots from the red plots is that for the red scenarios here, scenarios 2 and scenario 4, I have incorporated a high percent of donor variability. So in scenario 2 here, my donor correlation is 0.97. Again, your correlation will go from -1 to +1. Here we tend to have correlation that's positive. Correlation here is 0.97, so almost equal to 1, which would be a perfect case. This one down here, it wasn't quite as high, it's 0.77. But what you see in the patterns on the right that make them distinct or different from the left is that these tend to be parallel. Okay, so you can see that the donor here who had the highest value among the 6 donors in the reference group also had the highest value over here. These profiles tend to be parallel. And so you don't have any change in ranking of the donors on the attribute. And you can see again down here, they're parallel with a slope. Here they're approximately parallel and they're flat. Contrast that to the case where we don't have correlation and they all look like spaghetti strings that you just drop on the ground here. Here, the donor that had the highest viability in the reference had the next to lowest in the post change. So again, this is indicative of an attribute where we do not have high donor correlation. What that means is percent of total variation is a very small percent due to donor. These have a very high percent of variance due to donor. So what's called the interclass correlation is how we quantitatively measure that fact. And what I'll show you is the power curves depend very much on whether you're a blue or you're a red. And this is one of the problems you can run into when you design these studies, is that some of your critical quality attributes will be blue and some of your critical quality attributes will be red, will have a high degree of donor variability. The red ones are going to have very good power for sample sizes as small as 3 donors. But what happens is for those attributes that are like this, you don't get that power bump. And if you use less than 6 donors, as I'll show you, your power curves are very low. You have a very difficult chance of demonstrating equivalence. So visually, that's kind of the message of my talk here. Probably the best, in my opinion, the best reference guide we have for comparability studies in CMC comes from the EMA 2021 reflection paper. And I have that referenced here at the bottom of my slide. Is that they frame this problem by addressing 2 issues. One is what's called the need to define a similarity condition. And this is an extremely important point. The similarity condition answers the question, what does it mean to be comparable? It's very hard to argue whether data sets are comparable if we haven't even defined what comparability means. And so the EMA recognizes this and talks about the need for the comparability study to have within it a similarity condition. Second, after you've defined the condition, then you have to have the criterion. What evidence do you need to declare that the data sets meet the similarity condition? And finally, in coming up with this criterion, it's imperative that you study the operating characteristics, or what we call the power curves, to ensure that there's a good probability that we will meet the criterion when in fact we should, when we satisfy the condition, and conversely, a low probability of meeting the criterion when we don't satisfy the condition. So 2 key concepts here. And one of the approaches I've used that I think works well in defining the similarity condition is process capability, something we're all familiar with in the CMC world. So by process capability, what do I mean? Well, if we take a look at my reference distribution, I like to think of it as a manufacturing process here, based on a normal curve with red lines at 3 standard deviations from the middle of my distribution here. So I view these as 3 sigma specifications. Now, as you go through these calculations and you're working with processes that are established and you know what specifications are, they can always work into this calculation structure. But I'm doing that from a very generic situation that you can use in any situation in my mind. We just envision our reference group as a process that's centered between 3 sigma specifications. And as we shift distributions, as we look at different post-change distributions, we align where they are relative to the 3 sigma specifications that I've set up on my reference group. So in this case, in all my examples today, I'll assume equal variances between the 2 distributions. As I shift my distributions, my post change more to the right, more of that distribution falls out of specification. And this is what we refer to as a capability measure. Capability measure is the ability of our process to stay within our specifications. So as we become more dissimilar, then our OOS rate increases, or I just talk about the capability as the percentage of the distribution that falls within the specs. So the way that I quantify these visualizations is through what I call the effect size, or as everyone calls the effect size, which is the difference in means between the two distributions, the reference and the new, divided by the standard deviation of the reference. This is what's called the effect size. So looking at my pictures again, my dissimilarity will get greater as the effect size goes up. In these pictures, I start off with a mean of 0 and a standard deviation of 1. So if I shift it 1 standard deviation, that would be 1 unit. So my mean increases from 0 to 1. The shape remains the same because I'm not altering the standard deviation. So this has an effect size of 1. Here I've moved it 2 standard deviations to the right. It has an effect size of 2. And here I've moved it 3. Clearly, a process, the 3 standard deviations from the reference mean, has a 50% OOS rate. I think most all would agree that this process is not comparable to this process. That's quite a shift in the capability. The trick is trying to figure out where in between here we want to land when we define comparability for our similarity condition, this border condition that EMA recommends that we define. So here's an example, and I'm going to use today the effect size of 2 as my comparability or similarity condition. So here's what it looks like visually. The blue distribution is my reference group, and the pink is my post change or my new one with an effect size of 2. So I've shifted the mean 2 standard deviations. If I calculate the percent of the pink curve now, that is to the right of the 3 sigma specification here, you'll find there's 84.1% of the total area of the pink within the specification. So this would translate to an OOS rate of 16%. For my examples, I'm going to refer to this as the capability, the 84.1, and this is going to be what I call my boundary condition or the similarity condition that I want to establish. Here's a picture then of my 3 curves with the capability measure, an effect size of 1, 97.7% of the new distribution falls within the 3 sigma specs. Effect size of 2, this is the one that I just mentioned, 84.1%, and the effect size of 3, we have the full 50%. And I have a table here just so you get a feel for how fast these change as you move out on the distribution. So you can see here a 1/10 of a mean shift close. You don't see much change in capability, but as you get more extreme, a change of a 1/10 tends to get different pretty quickly. We're changing in terms of 2. So the issue is, what do I define as the boundary condition? So the MA document says, first, define what you mean by comparability. So what I'm saying today is this is my definition of comparability, that there is 84.1% coverage, if you will, of the new distribution of the area in the pre-change distribution covered by its 3 standard deviation specs. So why is that? So someone would say, well, Rick, 84% is pretty high. No one would accept a process with 16% OOS. And I would agree with that. So this is the way I look at it. And these quotes are mine. It's not from the EMA guidance. I believe a similarity condition should represent something that's clearly unacceptable, okay, as opposed to marginally acceptable. because the reason is when you compare, when you do your equivalence test or any kind of test, you're going to have a low type 1 error rate. The type 1 error rate is the probability that you will claim equivalence at that boundary condition. So I'm going to have a very low probability on this event I call clearly unacceptable. And for that reason, I think I don't want to have something that I consider marginally acceptable and put a very low type 1 error rate on that. So that's my argument. So I would say something marginally acceptable would be 95%. Something clearly unacceptable is 80%. So I would expect to pass the equivalence test a very low percentage of the time if I'm clearly unacceptable, and a moderately high percentage of the time if I am marginally acceptable. Okay. The other thing to keep in mind, again, are the sample sizes. All right. You may not be able to have a really tight capability measure if, practically speaking, we can only have small sample sizes. And in the split apheresis studies, most of the studies I've seen have between 3 to 6 healthy donors. I've heard rumors of as many as 8 donors in such a study, but given the difficulty of doing this, I don't know. I've heard that's as large as I've ever heard, but I've never seen them that large. But these are difficult experiments to run. So 3 to 6 is typically the sample size that we're working with. So the equivalence test of means is what I'm going to use here. And many of you are familiar with this. What we need to demonstrate is the difference of means of our two processes is less than the equivalence acceptance criterion, which is in effect the similarity condition. And then the burden of proof is on us to demonstrate this, and the regulatory agency controls the probability that we pass here with the type 1 error rate. So the type 1 error rate is the probability that we claim equivalence when in fact we don't satisfy the boundary condition. And the generally expected value for an equivalence test is 5% from the regulatory agencies. Okay. The decision rule then is you calculate a confidence interval on the difference of the two groups. This gives you a range of plausible values for the difference. And if all values in this range fall within the range from the negative EAC to the plus EAC, then you have demonstrated equivalence. How do we define the EAC? Again, if we use a capability argument, it's the effect size that defines our boundary condition times the standard deviation of the reference group, which includes variation due to process, analytical, and donor. So in my case, if my capability measure is 84.1 at the border, then my effect size is 2. My EAC equivalence acceptance criterion is 2 times the standard deviation. Okay, so let's take a look again now at my split apheresis design. And what I want to demonstrate is this equivalence test, the probability of passing that is very much a function of the donor variation. So this characteristic, this is what's called the interclass correlation. The correlation of objects or of values within the same donor is estimated by this Greek letter rho. It's the percent of donor variation over the total variation. So if donor variation, if there's not much difference from donor to donor, rho is low. And if there is a lot of difference from donor to donor, rho is high. Now here's an example of the equivalence test for scenario 2, where I had a high donor correlation. The reference standard deviation was 6.14. My EAC is 2 times that. So I have to demonstrate that all values in my confidence interval in absolute value are less than 12.2. Down here, you can see the interclass correlation is 97.4. It was high for that group. My confidence interval goes from -7 to 1.57. All of these plausible values fall within the range of negative 12.2 to plus 12.2. So this is demonstrated that these are equivalent. And remember, I simulated these two with the same average. So this would be my expected result. I've demonstrated equivalence of means here. The entire confidence interval fits between the range of negative EAC and plus EAC. Scenario 1, I also simulated with equal means. So we should hopefully be able to demonstrate this as comparable, but I have a low donor correlation. I have the same EAC here. I've got the same variation in my reference group. Here, my interclass correlation is only 13%. My confidence interval now is between negative 3.8 and plus 7.7. It still demonstrates equivalence because it falls within negative 2.2 to plus 2.2. But what I wanted to point out is, look how much wider this interval is than what I had in scenario 2, where I had a high correlation. This confidence interval is roughly 11 units wide, whereas this one's only 2.3 units wide. And because the EAC range here is fixed, the wider interval, again, has, it's more difficult to fit a wider interval within the range than a tighter interval. So the result of having a high interclass correlation that reduces the width of the interval makes it easier for me to fall within some fixed range here. That's kind of the lesson that I try to emphasize with my clients who are doing these experiments. Okay, so we've demonstrated that the type 1 error rate is set at 5%. There's only a 5% chance we'll be able to claim equivalence if the true capability is 84.1%. But what can we say about these conditions that we think are marginally acceptable or acceptable? What's the likelihood we're going to pass there? And to answer that question, and this is also highlighted in the EMA document, we need to use our power curves. So here's an example of a power curve for an equivalence test with 2 times sigma R is the criterion with 6 donors. On the horizontal axis, I've got the process capability. On the vertical axis, this is the power. Power is the probability of claiming equivalence. So you can see if I'm at my boundary condition of 84%, regardless of what the correlation is, or regardless of the sample size even, I have the patient risk or type 1 error rate set at 5%. The red dashed line is scenario 2, where I have the high interclass correlation. The black line is scenario 1, where I have the low interclass correlation. So for example, on the black line, if I think 95% is marginally acceptable, reading vertically up to the black line and across, you can see the probability of passing there is only 30%. There's an only 30% chance you can pass if the capability here is 95%. Instead of 95 to 100%, I think most people would argue, yeah, these are comparable processes. Their capabilities are very close, and yet you'll have very low probabilities of passing there with a sample size of 6 when the correlation, the interclass correlation is low. If you're lucky enough for an attribute to have a high interclass correlation, then you obviously have a really nice power curve. And if the capability difference is 95%, you're virtually sure to pass that. In fact, here you're virtually sure to pass that all the way down to a capability of 90%. Now here's the curves that give you power curves as a function of rho, okay, based on the sample size that you use. So I start with 3 donors. And I've seen companies start off, they think they want to use 3 donors for this kind of study. If you have a high correlation, this is the dark black curve here, a 90% interclass correlation, yeah, 3 donors probably gives you sufficient power. At 95% capability, you have an 80% chance of passing. But if you look at the case where you have less than that, at 50%, if they're identical, you only have a 50% power. If the interclass correlation is 10%, you only have a 30% chance of passing. And you can see these go up. The green lines are the ones that I chose for my example at 6, where even with a low interclass correlation, you still have an 80% chance of passing when the 2 processes are identical. So what do you do? So like I said, in many cases, you might have 10 to 12 quality attributes. And it's not unrealistic to believe that, just on my experience, of the 12, maybe 8 of them have low donor variability. So you have a lot of quality attributes where your power curve is really not sufficient. So what do you do in these situations? Because getting more healthy donors in the study, as I said, it's a very difficult process. So it's unrealistic to think you just simply increase sample size. It's often not practical. You can change your alpha value, but regulatory agencies seem to like 0.5. You can change your EAC, but getting much above 2, you'll probably get pushback. So what do you do? So what I recommend is leveraging. In most cases, you have other pre-change lots, independent lots. And if you leverage those and combine those with the split apheresis lots, then you can boost up your power. So here's an example where I have 3 paired lots. That's the dark black. I have increased the power by adding 7 independent lots that they had of other pre-change process lots. And I move from the black to the dashed line. So that's generally my recommendations, that if their sample sizes are in the 3 to 6 range, 6 is better than 5, 5 is better than 4, 4 is better than 3. But considering practical situations, if you're in that, then take your independent data from the pre-change and leverage that by placing that into the group. Okay. And
So
I think
Rick, I'm just going to do a
yes.
little time check here because we only
Yep.
have 3 minutes left and we have a couple questions in the chat.
Well, I thought the questions came on the panel, but I'll go ahead and stop. This is my final discussion here is discussion with the regulatory agencies is critical. Defining comparability provides a framework for the discussions, and you should use power curves to facilitate it. And both parties need to understand what are really practical sample sizes. So I'm sorry, I thought the questions were going to be in the panel.
I think most of the questions are going to be in the panel, but we had one question up in the chat.
Okay. All righty.
Good question. The important advantage of this approach is that it proposes a revision in the usual equivalence approach by treating the reference group as a kind of external standard. Can you comment on that?
Well, again, from a statistical standpoint, that's true. The external standard is the donor. Each donor becomes a standard for themselves in a pair design. So again, in statistical situations, this idea of matching on donor is a good idea. But again, the efficiencies only come if that blocking factor, if donor here, contributes to the total variation. Because when you calculate now the confidence interval, you've removed from that interval variation due to donor.
Thank you.
Okay.
So we have just 1 minute left. Do you have any final comments? We can save the rest of the questions until the panel discussion.
No, I think that was mostly what I wanted to do.
Great. Thank you, Rick, for a great
All right.
talk.
Yeah, thanks for your attention.
All right. So I think we have another scheduled break here, but maybe we'll just go ahead and start with the next session. And then we'll take a break before the panel discussion. Does that sound okay for everyone? I'm going
Yep.
loose and fast.
Yes, it sounds fine to me, Joanne.
Okay, great. So our next speakers are John McMahon, did I get this right? From Humacyte Global, and Catherine Gicoletti from Merck, and they're going to discuss Bayesian modeling to demonstrate comparability in a novel clinical stage bioreactor system for manufacturing tissue engineered human acellular blood vessels. So I'll let them take it away.
All right, and are you seeing my screen now?
Yes.
Perfect. Let's see if I can even get it into presentation mode. And sorry about that title. I know we gave you a bit of a mouthful, but at least you saved me from having to say it now. So I appreciate that.
Much appreciated.
Is everyone seeing that? I'll take that as a yes. Otherwise, give me a shout if you don't. So I wanted to thank the organizers for giving us the opportunity to present some of the work that we've been doing at Humacyte, and to thank Catherine for also reaching out to me and thinking that we were interesting enough to come and participate in this event. So it's my first time here, and I'm not a statistician either, and I'll try and say that without smiling. All right, so the obligatory disclosures. I am a full-time employee of Humacyte, and Catherine is a full-time employee at Merck. Just really briefly to tell you what we're going to be running through today. I'll start off by giving you a little bit of the clinical history, what the problem is that we are trying to solve here at Humacyte. Talk a little bit about how we go about making our product, which is a human acellular vessel, or an HAV. Set the groundwork for a problem or a challenge that we encountered during the course of our clinical trials, arising from an increase in the clinical demand for our product and a need to scale up our manufacturing facility according to that. And then I'll hand it off to Catherine to talk about some of these challenges and the statistical approaches that we're able to use, and how we apply them in this particular scenario. So starting off with the clinical problem, what we do at Humacyte is we make essentially a fancy tube, and there are a number of different reasons why you would need a tube like that. And essentially this is to do the plumbing in the vascular system. If you have an artery or a vein that needs repair, or an arteriovenous access constructed, there are a number of different conduit alternatives that you could choose to accomplish that goal. And just listed out here are some of the needs that we're studying and the different phase that they're in. So we do have a couple different clinical programs that are running right now. These include hemodialysis. So this is constructing an arteriovenous access to remove and clean blood for people that need to undergo hemodialysis. There are vascular trauma events where we need to come in and do an arterial repair of an injured vessel. Peripheral arterial disease, so a chronic condition that's going to necessitate an arterial reconstruction as well. And I think a lot of people are probably fairly familiar with CABG, or coronary artery bypass grafting, and why there's a need for that clinically as well. And maybe people might be less so familiar with the BT shunt, or the Blalock Taussig shunt, which is essentially a vascular reconstruction that happens in the cardiopulmonary system, and it's for a congenital condition, essentially blue babies that are not getting enough oxygen. It's a method that's done, a procedure that's performed to correct that and to allow proper oxygenation of the blood. And I'll say that in all of these cases, there are situations where there are significant unmet needs from any of the products that are out there as standard of care right now. So it'll depend a little bit on the indication, but oftentimes the use of autologous vein is a common procedure, but there's not always autologous vein available. And as you can tell, there were a couple different indications that were listed there. If autologous vein had been previously harvested for one of these indications, it might not be available for another. So there is a significant shortcoming as well. Synthetic grafts are another alternative, but they've been borne out in the literature to have some shortcomings, particularly in areas where there's a risk of infection. So there is a need for a novel conduit to perform these different arteriovenous reconstructions and repairs. So with that said, I'll just quickly describe the product that we make and the manufacturing platform that we use to do that. So like I said, we make essentially a fancy tube, and it's fancy because we do that starting from a working cell stock. So we have harnessed donor cells from tissues in the past that we now have banked in working cell stocks. We can take these cell stocks, expand them, and then grow them in a 3D culture system where we are able to manipulate the system in a way to encourage them to secrete extracellular matrix. And then finally, we're able to actually wipe out the cells, leaving behind just this extracellular matrix. So what we end up with is essentially a blood vessel scaffold that we're able to go and use for the surgical interventions as required for the indications that I was just talking about on the previous slide. The reasons why there may be some benefit to this process for making a conduit rather than using a synthetic conduit is that we have cell-derived matrix that's comprising the HAV, the vessel. And what this allows is it allows host cells to integrate and to remodel the vascular scaffold. And we think that it's possible that this will convey improved infection resistance, because this is one of the natural processes that have to occur for blood vessels to fight off infections, is to have these host cells be able to integrate, survive, and function and fight off infections. And it also conveys the ability to self-heal. There have actually been some instances where we've been able to see that after our vessels have been implanted for some time, they can actually bleed when they're cut. So they're really functioning in a similar way to a native vessel. So I had mentioned previously that we had a number of clinical trials that had been ongoing, 3 phase 3 trials at one point in time, and the technology had started as a single glass bioreactor that was able to generate a single vessel. And over time, we've had to have multiple evolutions and iterations of the manufacturing technology to meet the demand that we were seeing from increasing the clinical trials. We were at a point recently where we were moving towards a system that was able to grow 10 vessels at a time to a 200 vessel platform. And that's what you're seeing in the slide here. So this is where we well, one of the times that we interacted with Catherine to identify a solution to a problem. And the solution was that as we're scaling up this manufacturing platform, we know that there are going to be multiple sources of variability that are introduced into our manufacturing system. And it wasn't obvious to us that there was an easy way to do this using traditional frequentist methods. So I think at this point, I'll hand it off to Catherine to talk a little about the Bayesian methodology that was applied for our particular problem.
John, I'm gonna switch and share just so I don't have to ask you to click through my animation. So, and then I'll switch it back to you. Let's see. And Go into presentation mode. All right, so everybody should be seeing the same slide John was just on. Yes? Take that as a
Yes.
yes. All right. So yeah, so as John said, there are some challenges with a comparability analysis here. One being that there are multiple sources of variability acting on the process, and there are different multiple sources of variability in the two processes he described. The earlier phase studies were done in, or the late stage clinical studies were done in a process that looked essentially like this. It could make those 10 vessels per batch in a single drawer. And the commercial scale process was going to be able to make 200 vessels per batch broken up in drawers within cages. And there were some parts of the process where, you know, we wanted to make sure that CQAs might not be affected by variability from drawer to drawer, cage to cage, even though in essence, if you go back, you know, you're taking a drawer and just replicating that in the larger process. There are, you know, there are materials in the process that are flowing through these drawers and cages. And so there, obviously that's one of the things you want to show in your comparability, is that scale up isn't affecting CQAs. And we know that patient risk is linked not just to differences in means, but to differences in variability, which is why these different sources of variability in the process are relevant, right? Our typical approach of comparing means for comparability doesn't really address the full question about variability from a patient risk perspective, because it's focused just on the means. And while yes, statistical comparisons of means will be penalized at some level if there are differences in variability, it's not directly addressing questions about differences in variability. And we also know that, you know, when we think about the impact of differences in variability on patients, when we talk about variability, it's the cumulative effect of these sources of variability that leads to differences in variability overall. And so if the sources of variability are different in the two processes, which we know they are here, it's critical to evaluate both the, you know, relative contributions of those sources of variability and the impact they have on total variability and ultimately on patient risk. And so these are the challenges that Humacyte was facing in trying to design this comparability exercise. And so we discussed, you know, how to do that. And Bayesian prediction seemed like a very suitable, and honestly, even though, yes, the math is quite complicated compared to a frequentist solution to this, like a very straightforward way to do that. And why do I say that? So I'll go into a little description of Bayesian. Many of you have seen this quite a few times before, perhaps, or do this, but some of you may have not. So in Bayesian modeling, we describe all of our parameters, a mean or a variability estimate, with a distribution. And those distributions, let me put my pointer so you can see it. Those distributions describe plausible values of the parameter. So this one might be the overall mean of either the pre-change or the scaled up process, right? So the mean of a CQA from one of those processes, instead of just having a point estimate, we have a distribution of plausible values for that parameter. And that the width of that distribution reflects statistical uncertainty about the parameter. And we also have distributions for every one of those sources of variability. So the variability among drawers in the scaled up process, the variability among cages, the variability among vessels in either one of the processes. And I didn't put that in the graphic, but some CQAs were measured multiple places on each HAV. So there's actually even another source of variability that was there for some CQAs in both processes. And so every one of those parameters is described with a distribution of plausible values. And so when we predict individual results, and I should back up and say the other thing that we really considered very carefully in this comparability exercise was in linking our patient risk to not just means, but to variability. Really what that means is that we were interested in the distribution of individual CQA values in looking at comparability, not just means, right? And when we want to predict future individual values from either process, we can go and look at these distributions of parameter values, means, and variability sources, and pick a plausible value from the distribution and predict a new individual CQA value, CQA measurement. And then we go back and we pick another plausible value from the distribution and predict a future CQA measurement. And we do that over the full distribution of those parameter estimates, and we're going to choose the more likely based on our data and based on our model, the more likely values of the parameters more often than we are the less likely values. But we're still going to consider those less likely values. And this is what makes this, for those of you who are statistics geeks, this is what makes this importantly different from bootstrap simulation, right? I'm not just taking a point estimate of the mean and variability estimates and simulating new data. I'm considering the full model and database range of plausible values of each of these parameters and predicting new results in the future. And those distributions are weighted by the likelihood of those parameter estimates based on my model and what I know. This modeling was done in a non-informative way. I didn't use any prior information in this model. And to me, really this prediction, the ability to make this kind of prediction is what makes Bayesian modeling in a CMC space so incredibly valuable so much of the time. We have lots of sources of variability. We're very interested in individual value distributions, not just means. We know that, or at least we think that, we're going to have a hard time arguing about using prior information, but I don't need it to get all this benefit, right? Yes, sometimes it's quite beneficial, and I think we should keep making that argument. But this benefit is here regardless. I didn't need prior to do this. So I can predict a range of future individual values of any CQA for the HAVs from one of the processes. And that's accounting for the mean and the sources of variability. And with that, I can do all sorts of things. I can calculate the difference between the two processes. I could actually look at, I could go back and focus on the means, or I can focus on a predicted distribution of differences in individual HAV values from the two processes and answer questions that directly address patient risk in my comparability exercise, while accounting for all the sources of variability that are not the same in the two processes. And that's in fact what we did. The other things we did to make this really patient-centric were to choose the parameters that were included in the comparability exercise based on clinical relevance. And they're listed at a high level here, the kinds of things that were considered the most relevant to patients, and they were ranked in terms of criticality, and then the both the sort of statistical rigor and stringency of the criteria were also ranked according to clinical relevance in this. So this was a very patient-centric comparability exercise, and indeed the comparability criteria themselves for this were based on clinically relevant ranges. So this is something we've talked about quite a bit in this conference, clinically relevant specifications. Well, this is comparability criteria, but this is a really nice example of where we were truly able to do this. Clinical outcomes and CQA measurements were available from the clinical trials for individual grafts from individual patients. And so the comparability criteria for a number of the CQAs were set based on what was known to be clinically meaningful to patients. And that's huge. We set those criteria based on what's important to patients, and then we go and say, and we're going to do our comparability analysis based not just on means, but on ensuring that there's a high probability that my new process HAV results will be within these ranges that I know are clinically relevant to patients based on my clinical trial data. So that's a very patient-centric approach to comparability. And one other thing I'll say about that, the way I just described that, high probability that the post-change, the scaled up process, individual HAV results will be within certain criteria. This is another really big advantage of using a Bayesian approach, is that we can make those kinds of direct probability statements because we have a predicted distribution of individual results or individual differences, however we set up the analysis. We're not limited to saying that a statistical interval is within or outside of certain limits. We can give a much more descriptive and much more nuanced statement, much more detailed statement. When we do the approach like a TOST and we say, my statistical interval must be inside or outside of some criteria to say something's comparable. If that interval is completely within those criteria, that's great. We know how to interpret that. If it's entirely outside those limits, well, we're not happy, but we know how to interpret it too. But what happens when it sort of overlaps? This leads to a lot of questions, a lot of consternation. Some people say, well, clearly you're not comparable. But we don't actually know what the probability is that that interval can span one of those limits, and it actually can translate to not a whole lot of risk from a patient perspective. What does it mean? Or it could mean a fair amount of risk. With a Bayesian approach, we can make this direct probability statement. What's the probability that a future individual result is within some limits, or that the difference between the two processes, whether that's based on means or based on individuals, whatever was determined most relevant for a given CQA, is within some limits. So it's another advantage of the Bayesian approach that we used for this comparability exercise. So with that, I'm going to turn it back to John to talk a little bit more about the outcome, some of which I was no longer there for, so he's more able to talk about that.
Yeah, of course. Yeah, so we did take some of the advice that Rick was giving, and I think I can back it up to it. And with some of these more complex approaches, I think it is important to interface with regulatory early on. And we did that. We sat down in a type B meeting, in this case with FDA, to discuss exactly what the plan looked like and outline how we would use this Bayesian methodology to demonstrate comparability between our scaled up system and our previous manufacturing platform. At that point, they also asked that not only would we submit a comparability protocol demonstrating how different characteristics between the new and the old manufacturing platform compared, but also to supplement that with a clinical trial as well. So we did do a pretty small clinical trial. And then when these were completed and we were able to submit for comparability, we did have some questions that came back as well. So I would say we interfaced early, we explained the methodology, and then we had 2 information requests that we had to go back and forth on a little bit before being able to come to agreement, which we ultimately did. We did demonstrate comparability, and we've been manufacturing on our new platform since then. So I think ultimately we did have a positive outcome, but it does take a little bit more back and forth, I think, in communication and collaboration with some of these different types of approaches. And I think that's essentially it. Was there anything that I left out? Yeah. I think that's it.
We have just a little bit of time for 1 or 2 questions. Vanita?
Yes, thank you so much for such a great talk. I really found it very interesting. I do have questions, and pardon my ignorance. I'm not a very Bayesian person. I'm trying to learn and apply it. My question is that to show comparability, did you still had to run certain number of runs at scale and at small scale? Can you maybe talk a little bit about how many samples did you have, or how many runs did you have at each scale, and then you were able to apply the methodology? I'm completely off. Please guide me there.
Yeah, sorry, John, I'll speak to that a little bit. So in this case, the HAVs from these processes were kind of to put it in layman's terms, sampled to death. We had a lot of data. I would say one of the things that probably that we could have had a lot fewer samples, but that was for a number of reasons. I think there was some overlap with PPQ runs, so there were samples being taken for PPQ as well. And so in this case, we didn't explicitly power the comparability study. The sampling was already done, and there was no question that we had plenty of data. In a Bayesian context, if you were going to go in and think up ahead about what is the minimum number of samples I need from the 2 processes, we don't talk about power quite in the same way in a in a Bayesian framework as a frequentist framework, but you are usually the word you're looking for just went out of my head. It's Monday, but assurance, sorry, Bayesian assurance. And so you're, because we don't do the null hypothesis testing framework that's used for frequentists, but you still want to have some idea about, under certain assumptions about what your data look like, what your outcome would be. But it is so you would look up where that's the kind of thing you would be looking for is Bayesian assurance, and it's typically you have to do simulations, so there's not going to be a straightforward calculation the way there would be for a frequentist approach. But I will say, in a situation like this, with so many sources of variability, I mean, this is true when I do PPQ things and other comparability exercises. Once you've got that many sources of variability in a number of batches, I've often found it to be the case that if you've sampled enough to be representative of all those things, you've got enough data for what, I mean, you want to check it, but often those are the things that are driving your sample size, even in a frequentist context, more than those power and sample size calculations, once you've done enough to make sure you represent all those sources of variability.
Because what I've found is that at scale, you can't have that many batches, so 3 batches at the most that they can manufacture. So I was trying to get some idea from you as to how many batches were you able to get at scale, and then small scale is possible, you can get 10, 15 batches.
Right, as the number of batches wasn't exactly what drove it in this case. I'd have to recall, and that was different for the 2 processes because they're so different in size.
Yeah.
The commercial scale and the small scale, but we were really focused in our sample size because we did have to provide some justification. Like I said, even though there were tons of samples, we did have to provide in the protocol justification for this sample size being sufficient. It was the number of HAVs, and the number of measurements rather than the number of batches exactly that was the justification there. But yeah, that definitely can be a limitation. But here, since our analysis was focused more at the HAV level, that's where we focus those.
Thank you.
Yeah.
There's a question and then a follow-up to that. What frequentist approaches were you asked to supplement with? And as a follow-up to that, if the approaches don't give similar results, do you believe they will accept the Bayesian procedure?
So we were asked to do some 2 one-sided tests of means, which quite unsurprisingly gave the same answers. I mean, the approach that we were using was so much more conservative than a 2 one-sided test of means, and included a comparison of means by, inherently in what we were doing. There was no real question that you were going to get the same answer in terms of what the means are. Now, obviously, depending on if you set different criteria, or you set, much wider criteria for the means and some really conservative criteria for individuals, could you pass one and fail the other? I would say the Bayesian here is the far more conservative. So the risk would be that we would have, once we had met the really conservative Bayesian criteria, there was really no question. And this was sort of the back and forth, was, look, you're asking us to do this follow-up analysis that is so much less conservative than what we did. So, okay, fine, we'll submit that. But otherwise, it's really, I think it's a case by case, and I've seen this in the clinical world. I can't remember how many years ago where we were submitting more conservative approaches for things that looked at variability and not just means, and had exactly the same conversation. Okay, but also do your comparison of means. And we said, okay, fine. And why are we doing this? Because it's what we're used to. But I think it behooves us as the companies to really be able to explain what we're doing, which is what happened here. Here's why we're doing what we're doing, why it's so relevant to patients, and ultimately, also supply what they're asking for, because it just wasn't that hard to do.
And one final
John,
question.
if you want to add anything, because like I said, I want, feel free, but yeah.
Nope, I think you covered it.
One final question. Are you able to share what CQAs you looked at? How many vessels from each process and how many patients? How did you derive comparability criteria? And this will be the last one before we move on to our next.
That one I'm going to let you start with, John, anyway, because I want to be careful about what.
Yeah, so I won't go into too much details on exactly what the tests that we were performing were, but they're along the lines of what you might expect from what we needed to get out of the vessel. I mean, there are certain arterial pressures that the vessel is going to be exposed to in vivo, so those all need to be considered. We need to make sure that we've thoroughly removed all of the cellular material, because this is what's going to be required to make it more universally implantable. So there are handling and sewing of the vessel in that. So there are criteria that we were able to consider that went into which CQAs. And in terms of how we originally came up with the criteria, I think we started by developing a target product profile, understanding what it was that we needed to do, and working through there. So I know probably like a little bit more detail, but was there a third part of the question too that I might have forgotten?
How many vessels from each process and how many patients?
Okay, yeah. So it ranged a little bit depending on the attribute that we were looking at for how many vessels. I think there were some that were up in the 900 range, and I think there were some that were as low as 8. So there was quite a range, and it's going to be dependent on which attribute that we were looking at. How many patients were in the trial? So I imagine you're talking about the clinical trial that we did to supplement the comparability report that we sent in. And I believe this was a 20 or 30 patient trial, and I can't recall exactly offhand, but it was right in that ballpark.
Great. Thanks, Catherine, and thanks, John. We'll save the rest of the questions to the panel discussion. And the next speaker is Jose Ramirez from KITE, and he's going to talk about minimum variabilities, a lower bound tail.
Cool. I had a question. So I'm not a statistician. I just had a question mainly for Richard Burdick or others as well if they wanted to chime in. So I deal a lot with there's different methods, different processes, or different instrumental measurement techniques, and wanting to ascertain whether these techniques or processes are similar or not. And I was wondering the definition of marginally acceptable as 84.1% with an effect size of 2. I was just wondering, is that a relatively established criteria for process comparisons, or is that just useful in only certain situations? Thank you.
So I gave that as an example today for this particular application. So in defining what you consider to be clearly unacceptable or marginally acceptable, it really depends on the particular application. And what impacts that decision is your sample size. Again, the risk that you want to assign for your type 1 error rate, and the risk as a sponsor that you're willing to assign to, that being the risk of not claiming equivalence when they are equivalent. So I think for most applications, the effect size of 2 on equivalence set would be on the high side. And I think for most of them, probably between the range of 1.5 and 2 is kind of where you're looking. That's why the power curves are important to understand what that looks like and what the probability is of passing for all your capability measures. And then I also want to throw out that this definition is, again, based on process capability, but scientists may have other more meaningful measures of what a truly impactful difference is for a response. So what I'm showing you is more kind of, again, an argument based on process capability, but there may be other scientific considerations that are more important than that. And so you can use that to define what your target is for the boundary condition and for the equivalence acceptance criterion.
That's great.
Yeah,
Thank you.
And in fact, I would even just emphasize the last thing Rick is saying a bit more, which is that, ideally those equivalence criteria should in fact be what is scientifically relevant. That's really the entire point of them. And sometimes we can use statistics and, you know, process capability approach or other approaches to help with that a bit when it's hard to define. But ideally, they should be based on what is scientifically meaningful.
Right.
And maybe just as importantly as clinically meaningful. And this ties in closely, and I think Rick knows this as you do, Catherine, that if we were to move to patient-centric specifications, we could then define clinically meaningful movements in the process distribution. Yes, I think having good process capability, Rick, remind me, is relative to the specification. And it seems like your
Correct.
approach defines a specification as a 3 sigma limit, not as a clinically meaningful series of limits. Am I wrong?
So, well, the kind of the generalized approach that I did today put them at 3 sigma, but if you have other limits, you know, the actual specification limits, you can put those in and redo the calculation. So mine's more of a platform approach and is used sometimes in situations when they don't have specifications, and yet we're trying to compare really manufacturing processes pre and post.
Great. I see another hand up. I apologize because not everybody's full name is on the screen. So I'll
see
start
2
with
hands.
Stina.
Stina,
If you're asking your question, Stina, we can't hear you. Why don't you go ahead and write it in the chat box and I'll read it out. I'm not really sure what's going on with the audio there. So how about we move to Jun Gao? I think we're having audio problems.
same thing, huh?
Yeah, I apologize to both of you. Go ahead and write your questions in the chat and I'll pick it up. Go ahead, Tim.
Yeah, you're stuck with me because of audio. This goes to Jose and Catherine, and I would hope that some of the other participants I see in the meeting might weigh in on this. I was kind of taken aback, Catherine, when you said you don't need a prior distribution to do this, and I'm not sure that's exactly what you meant to say. I think Jose characterized non-informative and or you know, other expressions of prior distribution that are closer to what you're saying. And this goes as much to the concept of this session, I think, is to prepare people to discuss these things in a consistent manner. I spent 2 years working with Dave LeBlanc and Stan Deming, and 95% of that 2 years was spent on terminology. We couldn't agree on terminology, and it was frustrating.
Mm-hmm.
So as we enter more expansively into Bayesian statistics, I hope we can sort of express things in a consistent manner. And I don't know whether I'm wrong, Catherine, that you're not using a prior, you know.
No, it meant simply an informative prior. So it meant that a vague or diffuse or non-informative, and yes, you see a lot of different words for this in the literature. But that all I meant was that we were not using an informative prior. Yeah.
But just as importantly, I don't know if people think that as we enter into new ways of doing statistics, you agree that we come up with a sort of a way of expressing ourselves. And maybe I'm going overboard on this, or maybe I still have a rash from my discussion with Dave LeBlanc and Stan Deming, which is more likely the case because it was quite controversial. And it was, as I say, a huge debate about terminology that just, and I kept making the case, we're going to be more successful if we can unify our terminology, because otherwise, when you talk to different statisticians, you're getting different messages. And as I say, I don't know, I'm not picking on you, Catherine. It was just a case study of where, you know, it may confuse people for you to say, I don't even have to have priors, than to say, as Jose has characterized it, you're using something, a certain kind of prior.
Yeah, and I mean, I think there's a certain value in terminology, but I think it's more important. I guess you do see a lot of different ways that that's described in literature on Bayesian. When I give this kind of talk, I usually do manage to use the word informative when I say no prior. So that was a slip today. You know, in communication with regulators and with scientists, of course, and in any publications about it, there's going to, you know, details about exactly what that vague, diffuse, non-informative, whatever word we want to use, prior was, you know, would be described in some detail. So I guess it depends on who you're talking to, because there's going to be much more detail provided in some contexts where I think, you know, what word you use for it may be less important than just making sure, as we would in any scientific discourse, that you can describe well enough what you're doing that someone else could replicate it, right? So.
Well, it's a clever way of not having to use the word Bayesian. And actually, I'm speaking to another, you know, case where people were doing modeling and simulation, which is practically speaking what you were doing. And they said, well, we're not doing Bayesian, we're doing modeling and simulation. And maybe the alternative, maybe the alternative is that if there is a shyness in the scientific community to working with people that do Bayesian analysis, we revert to a language that they're more accustomed to or more familiar with. You know, I don't know the answer. I'm just saying that, you know, as we advance with Bayesian, I want to get off the fifth circle is the problem, Jose. Anger.
You're projecting on us. But I think Paul is going to give the closing talk, and we're going to have a panel discussion, and he's going to address some of the Bayesian things, you know. But there is some suspicion. People have suspicion in terms of Bayesian techniques, and I'm calling these days the R&R. There is a resistance from the agencies. This resistance can be because they're suspicious, or because they don't know the methodology well, or because they think we're going to pull one on them and, you know, use some priors that are going to bias the results in our favor. And there is also the reluctance from the people in industry, because if you're submitting, you know, if there is a submission and your name is attached to it and you propose some Bayesian and that fails, then that's why, you know, people smile when they're not a statistician following that joke. So it's that piece that it will be helpful. Having these conversations is helpful, especially with people from the agencies.
Yeah, I would just say though that oh, sorry, Jose, that I think in my experience, most of this debate is among statisticians. I have had so few questions from non-statisticians about this, and we can run around in circles for hours, clearly, about Bayesian versus frequentist. What do we call it? But I would say modeling, if we want to talk about terminology too, modeling and simulation is not Bayesian. There are lots of ways to do modeling and simulation, right? So I just think, again, we want to be clear about what it is we're doing. We want to be able to describe it clearly and explain it so that it, not just so that someone else can understand it, but so that they can have confidence that we understand what we're doing and why we're doing it. And that's what gives someone confidence that it's appropriate and that we, you know, we understand why we should be doing these things. And again, you know, have I been burned a little bit in you know, exchanges about one type? It's not just Bayesian, other types of statistical approaches as well. We all have in submissions to the agency. It is not limited to Bayesian, but more often than not, you know, when we do get questions, it's not from the non-statisticians. It's from statisticians who, in my opinion, frankly, should understand what we're talking about better.
Yeah, but that's the thing that they don't. In a lot of cases, they don't, and it's agency dependent. I'm not going to mention any agencies, but we can rank them in terms of how resistant they are, and part of that has to do with how much do they know about these techniques. But I agree with you. One of the things that we always recommend, if you're going to use some type of prior, you have to put a lot of information as to why you chose that and all that. And what I tell people when I work with them, even if it's not patient, the things that we submit, they have to be defensible and reproducible. And part of that defense is all the detail. And also that influences people being able to reproduce what we do.
All right.
Yeah.
At this point, I'm going to ask that we move on to the next set of questions. I have questions in the chat that I'm going to try and go through, and then I see a couple more hands up. I have a similar question to the one already posed to Rick. I am surprised if 84% out of spec is acceptable to the authorities when they also demand a CPK above 1.
So again, the only response I have to that, again, I don't define 84% as acceptable. In fact, I define it as clearly unacceptable. But what I am arguing is where the boundary condition has to be, because the boundary condition will have a very low probability of passing given that type 1 error rate. The other thing, so again, you can tighten that boundary to something you view to be marginally acceptable. But when you look at that power curve, your probability of passing, even when the processes are identical, is going to be very low for the kind of sample sizes we work with. My whole condition is if you get pushback, you need to show agencies these power curves. They don't want to set you up for failure. But if you're working with necessarily small sample sizes, you can't have extremely low type 1 error rates at extremely tight boundary conditions.
Great, thanks, Rick. Can you, Catherine, this one's for you. Can you elaborate more on what acceptance criteria is used?
I guess so if you're asking specifically in the study we described, then the short answer is no, for both confidentiality and because I don't recall. As John said, he wasn't comfortable disclosing the specific CQAs, so then it's hard to get into what specific criteria there were. But they were ranked according to risk. He mentioned a QTTP kind of approach. And then also categorized according to, and some of these, it was linked to risk, but anyway, for some of them, it was determined that the most clinically meaningful thing was differences in variability. And so those were ones where the way we approached it was to look at the spread of results from the phase 2 studies. Again, we had data, quality data from specific graphs from patients in the clinical trials and say, okay, we want to ensure with a high probability that the individual results from the commercial process are within these ranges. And so it wasn't a difference calculation exactly, it was setting margin, it was setting boundaries for the scaled up process based on ranges from the pre-change process. And for others, it was more of either a ratio or an absolute difference in means approach if that was deemed, either from a clinical perspective. For some of them, the differences were really more about process than about clinical relevance. There were some attributes that were looked at in the comparability study that maybe didn't have as much clinical impact, but had just some process impact. And so for those, it was more of a analogous to TOST approach, but using a Bayesian model that accounted for all these sources, had sources of variability in the model, but the difference was focused on the mean. But again, I don't think it would even be really interesting to tell you numbers for what those criteria were. And again, it wasn't, but maybe the question is linked to this 84% or, how many of multiples of a standard deviation. Again, that is not the kind of approach we took. The approach we took was to understand what the ranges and the variability were in the pre-scale process and say what would actually be clinically important to detect in terms of a difference or a range of variability in the commercial process based on what they knew about impact to patients, and the criteria were set that way. So hopefully that answers the question.
To have a follow-on to that, Catherine, could a nested mixed model with tolerance interval for the predictive distribution be similar to the Bayesian approach?
So could, however, calculating a tolerance interval like that, especially for the scaled up process, would be quite complex because you've got, for some of these CQAs, up to 4 sources of variability to include in that calculation. And even for the pre-scale, again, I didn't put the graphic down this far, but I mentioned that for some CQAs, there were multiple measurements per HAV. So even though it was 10 HAVs per drawer, you've got multiple batches, and then you've got 10 HAVs per batch. And then if you've got multiple measurements per HAV, you've already got multiple sources of variability, variance components, in other words. Calculating frequentist tolerance intervals, once you get past 2 variance components, gets quite complex. So.
This one's for Jose. Is there no trend or pattern in the viability over the 7 consecutive days, since it's okay to have the same limit for all days?
Well, that's one of the things that we going to take into account when we model the covariance or the correlation between the 7. Remember, there were several assumptions that were made. So the first assumption is that they're independent and they're all the same. And then we sort of relax that and we manage that dependency as a day-to-day variation. But talking about a trend, yes, the only thing is that even if you want to model that trend with a spline or something, you only have 7 points, which is not enough to model that. So the way, or a surrogate way to addressing that will be with a 7x7 covariance metrics that relates the days, and hopefully if you do that, then it may be like an AAR1 type covariance structure where the covariance is decreasing at, it's high between 1 and 2 and 2 and 3, but by the time you get 1 to 7, then it's less. So that will be something to also explore that way. Yeah.
Great, thanks. This question's for Rick. Do you have any experience discussing with the agency about this method? For calculating that, I think the specification that you mentioned as plus or minus 3 SD of R material is the key factor. Is there any challenges for setting the spec, for setting similarity conditions? I know that this was some kind of example to generalize it. I just wonder that the 3 SD is some kind of consensus for all.
So again, there's no, so is there ever pushback? Yes. Okay. So all of the discussions I've had, and again, these type B meetings are really useful forums to have these kind of discussions and agree with the agency what the criteria is. Again, I think in any particular situation, it's going to come down to, again, a meeting of minds between the agency and the sponsor, based on what both consider a practical sample size, and then fitting the criteria, the boundary condition, so that we have, as best as possible, keep the type 1 error rate low to protect patient, but also to give the sponsor a realistic chance for passing or meeting the criteria when in fact that is the case. So again, my recommendation isn't, to be used in all cases. It just depends on, again, the size of your data that you're working with. I might also add, now, Catherine addressed this, and it's something important to understand. I personally don't feel that equivalence testing is the best approach for comparability because it does ignore the differences that you might have in process variation. So in particular, in the CAR T cells, what I've seen is that when you're working with changes to your vector, they oftentimes will allow you to perform what's called the quality range test. That's the terminology the FDA has used. And I personally think that's a better approach for assessing comparability because it is sensitive to both differences in averages and standard deviations. However, for this one particular CAR T design that I've talked about today, they seem pretty adamant in having you do a test of equivalence. And the wording I've seen them use is that they view that to be a rigorous statistical test. And so that's why I chose that as my topic today. It's just a request that I see a lot from the agency. And in defining boundary conditions for those, I've had cases where 2 has been acceptable. In some cases, they'll push back and tighten it up a bit. But it just makes the whole discussion in those type B meetings more informed if you have these power curves to work from.
Joanne, may I interject a follow-up comment / question on
Hey,
that?
Jeff.
What's up, Rick? How you doing?
Good.
So I was just going to offer that I think the suggestion you made earlier around inclusion of the OC curve has been an essential aspect of whether you encounter agency pushback or not. In my experience, and Rick, I'd be interested to hear your perspective, if you're able to provide something accompanying the comparability protocol, the OC curve that forms the foundation of your determination of your EAC, generally my experience has been there's been very little pushback from the agency in that instance. So I think that is a key piece of evidence that all of us should probably consider when working out our particular approach for whatever comparability / tech transfer problem we're facing. Rick, I don't know if that's been different.
Yeah,
in your
no, I
case,
agree,
but-
Jeff. Thank you very much, but I agree with exactly what you're saying there. It looks like an interesting book on roller coasters. To your right.
It's actually a roller coaster kit with I was working with the kids this summer.
Yeah, I was admiring your book selection too. And I have a question really for everybody is, so you've all commented on presenting this and, resistance or acceptance from regulators. But in my experience, it is extremely rare to have a CMC reviewer who is also a statistician. So
Correct.
My experience has been that the reviewers are, certainly highly trained scientists from one discipline or another, but not formally trained statisticians. And so I put a lot of weight on methods that are easiest to explain to a fellow non-statistician scientist. And I just wonder, what has your experience been in explaining a frequentist approach versus a Bayesian approach, and versus, I'll say something maybe that's a hybrid a bit, like a Monte Carlo simulation type approach?
Can we let our regulatory colleague who's had his hand raised for so long actually step in and say something? June, are you able to unmute yourself and speak?
June.
Oh, okay.
Yes, we can hear you.
Thank you. I just changed my headset. Something wrong with the previous one. I think the resistance is there. That's the reality. There are a couple of things, and I think I mentioned with Jose as well. One Jose give our center a talk about the CMC Bayesian statistics. So the one thing is people are scared of content with intensive statistical method. It's not just about Bayesian. It's about if you provided the CMC reviewers a modeling result method using, for example, mixed model, they feel scared. If it is mixed model plus Bayesian, they scared more. This is just one thing. Is about the comfort zone. They are excellent, biologists, excellent, doctors, but for not everybody could understand those, statistical methods. Especially some methods, even like quite complex, even to statistician ourselves. So that's one thing. Another thing is, I guess one approach is like we should try from industrial perspective in submission, provided you apply the Bayesian method in the field that there are no frequentist approach exists. So they're empty. There's no other approach available, but only Bayesian works. From there, we could establish the reputation. See, like a Bayesian actually works in some situation very well. If the frequentist approach the method already very well established in that field, and the patient has to compete with that. And from regular perspective, the reviewers always will ask, okay, because you have provided the result and from the patient, could you also provide result from frequentist approach so we could compare them? It's becoming like it's a double the workload for both of the focus from the industry and also for the CMC reviewers. Because the frequentist approach result is already become something like a golden standard you have to compare to. In that case, the patient's approach become less valuable. Yeah, given that said, I still could one Jose give us and also Frank give us a 6-hour presentation in our directorate about CMC patient. We still see a lot of valuable examples. Like we expect to see some kind of breaking through in the future. The patient statistics could really bring something really promising in the regulatory submission.
Yeah. So
because I'm glad to hear that. The way I think of it is it's another set of statistical tools, right? And I get frustrated sometimes at the either or, right? As if these are two diametrically opposed paradigms. It's another set of statistical tools. And as was just said, sometimes the only, or certainly a much simpler approach without as many assumptions and constraints as a frequentist approach in certain situations, like the one I described and Jose was describing and others have in previous presentations here. I guess, to address the question that Joanne asked, I mean, that Julie asked, we've commented on it a little bit already. I think we've all had pushback sometimes on statistical methods. I actually have had statistics reviewers, and there were statisticians, or was a statistician involved in the initial interactions with FDA on the comparability protocol that we described. It is somewhat rare, but it does happen. And in fact, sometimes, as happens here, it was changeover in personnel that I think led to some of the questions we had later on, because we had initial agreement on the approach that was pretty strong. So, but I think education inside FDA is definitely a good thing, because people aren't as aware of Bayesian or may have some misconceptions about Bayesian, just as in other places. We see it in our own companies, so it's nothing special there. But I will say that in terms of ease of explaining, again, I don't think scientists that we're communicating with who are non-statisticians, which to your point is often who's reviewing this, necessarily have a hard time understanding what we're talking about with Bayesian. Bayesian directly addresses the questions that they're asking in a way that frequentist methods frankly don't. And we gloss over that in our description of frequentist methods. And so Bayesian, I find, is actually easier to explain most of the time. So I'll let Jose jump in, and I know we have some other hands up. But
yeah, so I just want to thank June. So you people know there is hope with Health Canada. Fang Cheng from SAS, the main developer of Program CMC, and I gave 3 2-hour sessions on Bayesian in CMC to about 90 people, statisticians, biostatisticians, and quality reviewers in Health Canada. It was a great opportunity. I mean, that for me was a dream come true, being able to talk to the agency about these things. Going back to your question, Julia, yes, I have seen that, but my experience now in this new environment has been the opposite. One of the first projects I worked with was a specification update. And we worked and we sent a lot of detail, and they sent questions, the RTQ. And there were 6. The first 3 were statistical in nature. They went to town and went over the calculations, the form and everything. So the responses had to be very detailed in terms of the theory we were using and all that. I did that, and we got the approval for that. So that was good. With another agency was submitting a Bayesian limit, like the one I presented, not for the minimum viability, for something else. And we gave a lot of detail as to the choice of prior, non-informative and all that, and how the calculation, the algorithm I presented was in the description. This is how you do it, step by step. And it was fine. They didn't question the methodology at all. So for me, that was a shock. So maybe, the environment, the CAR T cell space, I don't know if the reviewers are different, but slightly different experience that I had before.
Thanks, Jose. I see some questions here, hands raised, but I have an older question in the chat. Let me read that first. John mentioned that he had hundreds of vessels from the 2 processes, but something like 8 patients. What was the primary statistical unit then for showing comparability? The vessels or the patients? And were multiple vessels implanted in each patient?
So I can address that. So because there's a process comparability, so the unit we were comparing was each CQA measurement, which, as I said, sometimes was multiple measurements per HAV. I think the question he answered about number of patients was about a follow-up follow-up study that was done after the comparability analysis, not about the number of patients that was in the clinical trials for the pre-scale-up process. And some, so I think in general, in the overall, all of the clinical trials, some patients received more than 1 graft, but again, that wasn't really, the analysis was focused on the individual HAVs, the quality measurements of the vessels themselves.
And another question is, Jose, what would the hierarchical model look like for the multivariate model? What parameters go into the model? What assumptions regarding hierarchical levels?
For that one, it's not the hierarchy per se, but it's the multivariate nature of the model. So then you're specifying a prior for the mean vector, so to speak. Let's talk about the multivariate normal. So we'll be specifying a prior for the mean vector and then a prior for the covariance metric. So if so the variation will be taken into account via the covariance metric, because each column of that or row will be one of the days. So in terms of the hierarchical model I used, it was very simple because it was just one random effect. So you have your prior and your parameters and the random effect and some hyper priors on the parameters of that random effect. So it was simple. Nothing like, I can really imagine, Catherine, your model. I don't know if you want to comment about how complex your hierarchy was, especially now that you mentioned that you have repeated measurements too. So
yeah, I mean, it was pretty much, I don't have the slides open anymore, but I described on the slides, right? That kind of graphic is really useful for picturing those hierarchical models, right? Of batch and then the more complex model, the scaled up one, you batch and cage and drawer and HAV, and then I didn't put below that. Yeah, for some of them, there were repeated measures per HAV. So yeah, the sampling wasn't of each and every one in each and every drawer and each and every, so it's somewhat complex in terms of nested and crossed and everything, but that's a picture of the model in a sense.
There's a question for Rick. This is the final question in the chat. Rick, the error term you're using looks like it's the patent by process interaction. Is that correct?
The patent by process interaction?
That's what the question
I think
is.
patient.
patient to be patient.
I'm sorry.
Yeah.
Oh, patient. Yeah. Okay, that's correct. I mean, you assume there's no interaction and then that estimates your analytical error. Correct.
Apologies for the confusion.
No problem.
Okay, we're going to go to the hands. We'll start from my left to right. We'll start with Tim.
I hesitate to come back to a question.
No projections, please.
No, no, no objections, but I actually want to come back to the question of melding frequentist with Bayesian methods. And it actually, I think, came up in your talk, Catherine, where you said when you did the frequentist analysis, you got the same result. I think we deny that we don't recognize the fact that the likelihood is a choice of distribution. So, everybody who's afraid of picking a prior distribution, they're sort of fooling themselves because they've already picked a distribution. Therefore, I wonder that and this is another thing I learned in my 2 years with Stan and Dave, is you will get the same result under certain conditions, under certain distributional assumptions. And I wonder if we're making a big deal out of Bayesian simply over the matter you last mentioned, Catherine, which was, from a philosophical point of view, the Bayesian method answers the question you're interested in. But if you get the same result for the choice of distribution you made for the frequentist approach, doesn't matter.
Well, so I think what you're referring to is if you use a non-informative prior, and you'll get this, you should get the same point estimate. If you calculated a simple confidence interval or prediction interval, you'll get the same bounds. If you had a single variance component tolerance interval, you should get the same bound. But there are some, so there's the philosophical difference about the interpretation, which I do think is very, very important in terms of being able to answer these questions. I can make probability statements, direct probability statements about the thing I'm interested in, instead of about the data I collected under repeat sampling under certain assumptions about the process, which is what we're doing with confidence intervals. And when I say it's not as easy to explain frequentist as we say, that's why, right? But again, and to Jen's point, when there are some things that a Bayesian model can simply do more elegantly, or do, period, right? Like this extremely nested, and like I said, it's not just nested model that I ran, or the multivariate models with some nesting, and so there, when you say, are you going to get the same result, you may not even be able to do it frequentist.
That's right. Rick, can you derive a frequentist solution for multiple nested variance components? So
you can run a model. Sorry, go ahead, Rick. You can run a model, but in terms of calculating some of these intervals and the probabilities is what I'm talking about.
Yeah. I'm just wondering if, again, there is a frequentist counterpart, even though it's a complex nested variance component model.
Yeah, so here's my take on the whole thing, because I can even cite a more simple problem that I've run into with agencies, and that's the issue of transformation. Okay, so you have data often, as you know, Tim, in the USP, when you're working with titers or potency data, we like to work in the log scale. But I've had many occasions where I've analyzed data for a regulatory agency in a log scale, and they request that we also analyze it in an untransformed scale. There's this kind of, I don't know, thought belief that if you can come up with the same result in a manner someone else would have done it, then it kind of gives more credibility to your new approach, which I don't think is necessarily a good scientific way to proceed. But I think the point to be made is, whether you're using a Bayesian approach, whether you're using a transformation, you have to explain it to the agency in a way that makes scientific sense to them. So I think, to Catherine's point, oftentimes with non-statisticians, maybe the science makes more sense to them, but you have to provide that information in your filings and in the work that you do to prepare for these type B meetings, so that they understand what you're doing, right? That's just been my experience. So yes, I could, work on a similar problem using solely frequentist type approaches. And yes, in many cases, I know I've done some work with what are called generalized confidence intervals, which are computer intensive approaches, and they're really based on Fisher's fiducial intervals from eons ago, and show that if someone's using a Bayesian prior that's not informative, we come up with like almost identical solutions. I actually think there's a theoretical link there. So you can, but does that validate the Bayesian approach? I don't think so. The Bayesian approach has to stand on its own, and its goodness and usefulness isn't going to be determined, in my mind, by whether a frequentist approach can get the same decision.
Well, I don't like the validation. I do like the expedience. So if there are people resistant to Bayesian, the question is, does it make any difference if you took the pathway of a valid frequentist approach, other than you're not deriving the distribution? You again, at the end of the game, you're answering yes or no. It's binary. Yes, they're
Right.
comparable, or no, they're not comparable.
And what's, yeah.
So
But what
you
the
use
question
expedience.
is important too, right? And I think
Well-
that's what I hear from Catherine and Jose. In some cases, the question cannot be answered with a frequentist approach. So
Mhm.
the frequentist
Exactly.
approach is an expedient because it doesn't answer the question.
Yeah. I mean, take the example I presented. We can probably try to figure out what a lower tolerance bound for that is, but
Mhm.
you know, probably Rick can do it because he does, you know, he's consulting
I'll
now.
slap
but we don't have
together
time.
some Satterweight approximations
Exactly. It's
and
something
pull my
like
way
that.
through it.
Yeah, but there are things that you cannot do. And the other difference is, you know, you start thinking in terms of distribution. In the plots I show, if we get a lower bound, forget about the minimum, just a lower bound, you get a number. Okay. Here you have a distributions of lower bounds, and you're picking the 5th percentile. But now you get a sense for that variation. Is that distribution fat, skinny like me? It tells you a lot about, you know, your data. And we also forget about the sample size. If you have sample sizes that are huge, then they're going to overpower the prior, you know, unless it's a very informative prior. But so that also comes into the picture. So yeah, I'm with Rick. It's not about validating the Bayesian method. Is they stand on their own, and they allow us to do things that we may not be able to do at all or quickly with the frequentist approach. Plus gives you a different perspective. It's a matter of interpretation.
So we have some hands up. Can I go to Tara next?
Thanks, Joanne. Yeah, I'll just reiterate what I just heard Jose say. It's pretty well recognized that Bayesian approaches can answer questions that we just can't answer with a frequentist approach. It might not be possible, and if it is, the solutions aren't readily there like they can be with a Bayesian. But that's not what I wanted to share with the group. What I wanted to share with the group was I actually just came from the International Society of Pharmaceutical Engineers annual meeting. And Peter Marks, who is the head of CBER, gave the closing keynote. And his keynote focused on cell and gene therapy. And he explicitly called out the need for more advanced methods, such as Bayesian, to solve some of the issues we have in cell and gene therapy. He was mostly focusing at that time on the idea of small sample sizes and use of prior knowledge, for instance, use of similar drugs in the, you know, in a similar within the cell and gene therapy platform that might be similar to the product that's being developed. And so you could use that information as prior information and combine that with the very small sample sizes that you're limited to in cell and gene therapy to get better answers faster. So I found it quite interesting for Peter Marks to explicitly call out a statistical method in his talk.
Yeah. So
and how we have to, and we have to start leveraging leveraging these sort of methods.
Yeah, no, I agree. For me, it's very reassurance to hear that. And people may remember the talk we gave last year at the IABS, and it was essentially doing that, using prior information of similar products or molecules to inform the calculations of a new molecule with a small sample size. So yes, it's exactly what he's advocating for.
So there you will have to focus more on your prior than you have with these non-informatives. You've got to use that prior and provide real information.
Yes.
Yeah, although in an adaptive sense, and I've worked on this within a CMC application as well, you can start with a very diffuse prior and then accumulate information.
Build it up, right?
Build
it up over time.
Yeah.
Yep.
Yeah. And I'm not going to deny it, Rick. When I heard you at the end of your talk, you proposed, what if we have a small sample size and we bring the other data in? I was thinking Bayesian. See, that tells you that the
right.
transformation is almost complete.
Right, right. I'm
was
always
thinking,
looking
oh,
for data to build that you have
yeah,
to bring in. You've got to
yeah,
leverage that.
exactly. Yeah, but I was thinking, oh, that's the prior information that we can use.
Exactly,
Yeah, yeah, yeah.
and that's good. And again, my understanding of clinical trials in stage 1, they use Bayesian models in that situation because of the same situation,
Yeah.
small sample sizes.
And actually, now that you mentioned that clinical trials, the FDA has accepted, adaptive designs in clinical trials. I just came back, Paul was there too, 2 weeks ago from the Base Pharma conference, and 80% of the conference was on adaptive designs and how people are using, Bayesian methods in that area.
Yep.
So it's a given now, almost a given.
Julia, I see your hands up.
Yes, maybe I can take us away from Bayesian for a bit here. Jose, I still have a question about the minimum of 7 viabilities. It's really interesting, but I guess what my real question is, why 7? Did you look at the frequency of, is there some kind of cycling? Like, why 7?
Well, it's just an example. I cannot go into more details just because we're going to get into the process and all that. But just the process of developing this, remember there was a little slide that I said we kept the cells happy, we feed them, sing to them, and blah, 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 so they can grow. That takes several days. And that's all I can tell you. I just picked 7
Okay.
to show an example. Yeah. 7
Thanks.
nodes, 7 days of the week. Before I can get in trouble with the lawyers.
Thank you. I understand.
Yeah, but you can, you can apply it to 3, to 10, to 12. The math is just change the exponents and multipliers. Yeah.
Yeah, thanks for sharing that. That's an interesting approach that I don't think I had, I just wasn't aware of that. So that's a good one.
Yeah. And that's because it comes from reliability engineering. They're always working with the weakest link, so to speak. So they use either the smallest one or the largest one. And some of the distributions they use, like the Y-ball or the extreme value distributions, deal exactly with that. So
the answer is it's just not 3.
When everything else is 3.
What do you do when you have less than 3? Is there any statistics you can apply to that?
So increase your boundary condition.
I see June's hand is up, so I'm going to move to June now, and I have a couple of questions in the chat box.
Yeah. So one question, actually, I think Jose mentioned we should try to use non-informative distribution. Since from regulatory perspectives, they may compare the result between the Bayesian result to the frequentist result. So, but I have a premature argument. Because to us, like the value of Bayesian is it could borrow some, information from prior historical data. So that means like seems like if we have a clear idea about a relatively clear idea about what the prior should be, we should always choose the informative prior.
Yes, if you have that information, yes, of course. Yeah. I was just being very specific within CMC. If we can put in a limit or something like that, we don't want to bring additional data and all that, then we use a non-informative or diffuse or vague prior. That's all. But if you do have that information, like Rick mentioned in his example, you're dealing with a sample size of 4 or 6, but you have some historical data and that you can bring that in. Or Tara mentioned that, we had that example last year where we had data for similar products and we brought that in as a prior. And as Rick mentioned, now you get into an informative prior and really had to work out the details and provide all that information.
Okay. So second,
sorry,
go ahead.
Go ahead,
I was just going to say, I certainly agree. And I think as Jose said, some of it is that it's expediency, right? It's hard to explain and we frankly fear resistance, but absolutely. And I think as an industry, we're going to have to face that and move in that direction for some of the needs in the future. So, yeah.
Yeah. And the other one is that I mentioned that too, and I know there's disagreement between the Bayesians and people in industry, but it gives you a level of standardization and consistency. When I use that catalog, if I'm dealing with a beta distribution, I go for that Jeffreys prior, and it has worked well for me for the type of things I'm doing. If I'm doing another distribution, I go there and see what they have, and it has been very helpful in the sense of having some type of standardization. And I know Paul will talk a little bit about, you know, these ongoing work with CMC and the agencies, and guidelines is not the right word because it's not telling people what to do exactly, but providing maybe Tim, what you were saying about the language, part of that consistency too, you know, that, you know, my hope is one day we have some type of catalog of priors for CMC that people have used and say, okay, if you're in this situation, you can try some of these, that both people in the industry and the agencies are comfortable, quote unquote, with them. So.
Okay, so one of my colleague, he's biologist basically, and he when he discussed with me, he said like, if you guys using the non-informative, you know, for the Bayesian approach, that means you guys did not really use too much information from prior knowledge. In that case, why don't you just stay with the conventional fragmentist approach? But I could not find a very straightforward answer to him. So what are you going to see, Jose, if like you to answer his question like that?
But Catherine, you're gonna address that. I mean, there's things that you cannot do. So go ahead.
Yeah, no, either one of us. Yeah, I mean, there are things that we, you know, that were presented here. There are, and Tara mentioned it too. I mean, there are analyses that simply cannot, or at least cannot without many assumptions and much difficulty, be done in a frequentist way. So the Bayesian modeling has greater flexibility for more complex models. And it also, again, some of it is this interpretation, directly answering questions of interest, which you may think of as a purely philosophical thing, but you know, in the example that I provided, it's the ability to say something like the probability that that an individual future vessel has a measurement within this range is, 94%, instead of simply my interval is inside or outside of some limits, right? So it's more information, using that word in a different way from our priors, more information in your statistical inference than simply I'm inside or outside of some criteria. So that's another really important reason, I think, because you get a distribution to describe your results, you can make these probability statements. And so that interpretation piece, I think, is really key. So the flexible modeling and the interpretation, I think, are big reasons, regardless of whether you use information in your prior.
Yeah, and that's it. I mean, those two things, the flexibility for me, some of you know my pet peeve of the over-reliance on the normal distribution, even with transformations. The normal is not that normal where I am. Everything is not normal. If you look at viability, transduction, CD3s, those are quantities between 0 and 1. And then you have a lot of quantities that are positive by nature, bounded below by 0. So if you're very close to 0 and you use a normal, you're going to get negative result, which makes no sense, or you end up chopping the distribution, which is not that great. But with the Bayesian approach, as long as you know the likelihood, you can write it down. You pretty much can use that. And, I calculate tolerance bounds and intervals for many different types of distributions, beta, gamma, log normal, the T distributions. I even let the Bayesian procedure tell me what the degrees of freedom for the TR and all that. I mean, it's the flexibility. Trying to do that from the frequentist point of view is difficult. It's a lot of effort that, people in industry don't have the time. So plus the
just a couple more minutes. I have a couple questions in the chat here, and I'll ask this one and then we'll cut it off at that point. When you want to borrow information from a prior using data from another product, what is your key parameter to say, yes, I can expect these two products were similar or could have been similar? Your key parameter to choose a similar product to borrow from.
You want to tackle that, Catherine? You want to say something and then I'll say something, or?
Yeah, it's a big topic for a couple of minutes. This, I don't know. It's a big topic because when we talk about borrowing information, we can do that in a lot of ways, right? You're talking about, we're describing, so you have a model to use to describe your process, and that model has a mean, I mean, in the simplest form, right? Say we have a mean and variability, and that's a very simple model. Well, are we borrowing information about the mean, or are we borrowing information about variability, or both? Right, and so first we have to understand, because that's where the prior is going to come in. It's really going to be prior information about these parameters in the model, sometimes even hyperparameters. There's a whole language for this. And there are lots of different ways to do it, even once we identify what that is. But in terms of your question is really, I think, at its heart about representativeness. And there are other words that we use for that in Bayesian world, but the concept is still very important. And so there are ways to do that in the Bayesian science, in the literature, and there are also, just as with any other analysis, scientific assessments of the sort of relevance and representativeness of that information. That's probably where more of the discussion is going to come in when people ask you about your priors. I'm trying to keep it kind of high level here in terms of, instead of getting into the real questions about parameters and things, but because it's a big question. Jose, if you want to add anything?
Yeah, no, the only thing I'm going to add is what you were saying at the end. This is where I rely heavily on the SMEs, the subject matter expert, to determine under what conditions those products that we're going to use are similar to this. And if you remember, I'm bringing back that talk because it's related to all this. Last year, the SME that approached us for that problem, she also took part in that presentation. And we talked about the things that they looked at to select those products that were used as a prior information. So that's key. You need that there. So.
Okay, well, we're right at 2:00. I'm going to ask everyone to thank our speakers and panelists, Jose, Rick, Catherine, and Valentina. And I think we're going to move on to the breakout session.